Good morning, ladies and gentlemen, here in Kuala Lumpur. Good afternoon and good evening to everyone, wherever you are. Now, this will be the first plenary lecture of our joint meeting of ISCE-APACE. Now, in this plenary, the first person I will introduce needs no introduction, actually, but I need you to make a guess. He's a chemical engineer by training, but he received his PhD in applied biochemistry. He has recently been elected into the National Academy of Inventors and made a new chair of the International Congress of Entomology Council, ICE Council, after the legendary May Berenbaum. And one more clue. He is an ISCE silver medalist, Silverstein-Simeoni awardee, and past president of ISCE. Ladies and gentlemen, may I introduce to you everyone, distinguished Professor Walter Leal from the University of California, Davis, USA. Now, Walter is going to present a lecture entitled Scuttle and Indole Sensing Odorant Receptors with Reciprocal Specificity Mediated by a Single Amino Acid Residue. Walter, the floor is yours.
Thank you. Thank you very much, Alvin, for inviting me to speak at the joint meeting of the International Society of Chemical Ecology and the Asia-Pacific Association of Chemical Ecologists. In case you don't know, the acronym APACE or APACE was first suggested by Tom Baker in a meeting in Shanghai, which I believe Alvin also attended. I apologize for not being able to join you all in the beautiful city of Kuala Lumpur. Please enjoy Alvin's hospitality and the hospitality of the people of Malaysia, not to mention the tour tomorrow. Alvin invited me to give a presentation, and soon after I accepted, he asked me to organize a symposium. His approach is known by the metaphor of carrot and stick. In case you are not familiar with this phrase, it refers to a reward and punishment. In this case, however, it turned out that Alvin gave me carrot and carrot, given that both activities have been very rewarding. I invite you all to attend the symposium Frontiers in Chemical Ecology, sponsored by BedoukianBio, which is going to start soon after this presentation. It will highlight rising stars in chemical ecology. Today I would like to talk about the two odorant receptors with reciprocal specificity to oviposition attractants. It turned out that a single amino acid residue mediates this reciprocity. This is the work of Flavia Franco in Ping Shi Shu for a few years in the lab, and a more recent collaboration with Vladimir Yarov and his graduate students Brandon Herrings, from whom I learned a great deal about membrane protein modeling and ligand docking. A manuscript from an earlier version of this research project has been submitted to bioRxiv, but today I'm going to talk about new and exciting results which have never been presented outside of the laboratory. It is part of a manuscript under consideration for publication. For us chemical ecologists, it's well known that mosquitoes rely on oviposition attractants to find suitable sites to lay eggs. Adult mosquitoes can explore new territories if they do not find what they need, but this is not the case for immature stages of mosquitoes. The mother must find places for the offspring to survive and flourish. In the case of Culex quinquefasciatus, the southern house mosquito, also known as the common mosquito, they make these beautiful egg rafts. 3 decades ago, Jocelyn Miller, who is in my cohort of chemical ecologists, discovered that the 3-methylindole scatol and indole are oviposition attractants for Culex quinquefasciatus. The results were soon corroborated by Blackwell and the others, as well by the field research by John Pickett and his collaborators in Africa. When mosquitoes OR started being deophanized, we identified CQR sensitive to scatol and indole. The scatol receptor, highlighted in this 2010 cover of the Journal of Chemical Ecology, is remarkably sensitive to scatol. The indole receptor was reported in +1 the same year. Since Ping-Shi Xu joined my lab 12 years ago, we have been so interested in studying the specificity determinants of these two receptors. It was not until Flávia Franco from USP, Brazil, joined the lab that this research got real momentum. Ping-Shi has completed the work after Flávia returned to Brazil and welcomed David to the family. CQR2, also known as CQR21, responds to scatol and gives a weaker response to indole. From here on, whenever possible in this presentation, all things scatol will be in red and indole in blue. It looks like American politics, red and blue, except that here in the presentation, the red is the good stuff. Contrary to CQR10, CQR2, also known as CQR121, is just the reverse. It gives dose-dependent stronger response to indole and a lower response to scatol. Of note, the currents recorded from the indole receptor are smaller. Shall we say CQR2 is the less sensitive receptor. Here we have the primary sequence of these two receptors with transmembranous domain predicted by Octopass. These transmembranous domains are highlighted in red, again for OR10, and in blue for OR2. These receptors, they have more than 370 amino acid residues with about 50% identity. We envision that generating chimeric receptors by swapping the transmembranous domains would lead us to specificity determinants. Please keep in mind that back then, there were no experimental structure for Durante receptors. We now have two, one of the core receptor orco and another one of the ANOA of a primitive insect. I'll get back to that later. The earlier predictions already point out that the binding pockets were formed by these transmembranous domains. There were even reports of one residue being imported for specificity, as the 2012 PNAS paper by Kevin Warner's group, for one example. After we have already tested about the 2 dozen chimeric OR, the groundbreaking elucidation of the first structure of an odorant receptor was reported by Roel, Butterwick, and collaborators. By the way, Joel will be here as one of the featured speakers in the symposium starting immediately after this presentation. As Frank Zuval put it, it was a landmark study. We then asked whether our TM predictions were accurate and whether we would have to discontinue or could progress with the work. As depicted here in ferrite, like kind of gray color, the predicted transmembrane domains by Octopass and the experimentally proposed segments overlap so well that gave us confidence that Octopass predictions were accurate and validated the results of our chimeric receptor test before the structure of ABAC orco was reported. Let me explain what we mean by swapping domains. We use the CQR10, the scatol receptor, as the recipient because it is more sensitive. Then we quote-unquote borrowed transmembranous domains for CQR2. In this cartoon here, I show an example of a chimeric receptor with 2 domains, M1 and M4, from OR2. We do not have a standard nomenclature for these chimeric receptors, so after discussing with Jason Pitts, we adopted the superscript M after CQOR10. Here we have CQOR10 superscript M1, M4. Sometimes it's difficult to write superscript in some slides, so forgive me for that, but M should always be subscript. Let me give you an overview of the years of research so that I do not go into details and leave enough time for questions and the answers after the end of the presentation. Here's a cartoon with two ORs, one specific to scatol, CQOR10 on the left, and see that the higher dose-dependent response depicted in red. The receptor on the right is for indole, and the stronger responses are elicited by indole with a minor response to scatol. Even run the risk of being perceived as provincial, let's look at the curve, the lines. On the top, if it's red, the receptor responds to scatol. We call it scatol-specific receptor because the other curve is so far below. I think I lost my credibility among physiologists for trying to make it so simple for a wide audience. We went on and produced chimeric receptors. They were specific to scatol as CQOR10 or did not respond. Then we find one chimeric OR, CQOR10 M2 M7, which reverse the specificity as shown by the blue line well above the red line. We note that every time we include the M5 or M6, we found a receptor with a much higher sensitivity. Then we try M2, M7 plus M5 and M6 under the hope of increasing the sensitivity of the receptor. Indeed, the chimeric receptor was more sensitive. I will get back to that in a moment. Lastly, we generated single-point mutations and identified a single residue mediating this reverse specificity. So let's go to the details here. Of course, the table is probably low resolution on Zoom, but we don't need to read anything of the table. Just look at the colors, and from the top you see OR10 in red, and all the other mutants which were either black, that means they did not respond, or red, that they respond to scatol. So let's save time in not going into details on those, but let's go directly to the one on the bottom. You see that one in blue. It shows reverse specificity. It displays specific response to indole in blue, and that's where I'm going to start and skip all the previous mutants. Again, CQOR10, M2, M7 showed a clear dose-dependent response and specificity to indole. As a reminder, here's a cartoon of CQOR10 so that you can compare the two responses. The current recorded from M2, M7 were small compared to CQOR10, so we included, I mentioned before, M5 and M6, which were nodes to always enhance, for some reason, the response of the receptors. Indeed, when we get CQOR10, M2, M5, M6, M7, the current were stronger than the current recorded only with M2, M7. By this time, we already knew that the M7 is not part of the binding pocket, as suggested by the publication by Butterwick's group, and more recently by Josefino de Marmol and colleagues reporting the structure of MHRA-OR5 from a primitive insect. By the way, that receptor is a standalone receptor whereas the OR from all evolved insect requires the co-receptor, ORCO. In the Frontiers in Chemical Ecology Symposium, soon after this presentation, Joel Butterwick will share his perspective on this non-binding subunit called ORCO. So, even though that M7 is not supposed to be part of the binding pocket, we tested CQ10M7. This mutant did not reverse the specificity. It remained specific to scatol, as shown here with the line, the red line on the top and the blue line on the bottom. We then surmised that M2 holds the key for the specificity. By comparing 3M2 primary sequence in OR10 and OR10 M2, 5, 6, and 7, we see that there are three segments that differ. We name them outer, middle, and inner segments. Again, we are looking only at the transmembrane 2 because there are 4 transmembrane that have been swapped there, but only transmembrane 2 is important in terms of specificity. And within this transmembrane 2, there are 3 parts that we could separate: outer, middle, and inner segments. So first, we restore the outer segment. We mutate again, start with the mutant, and mutate it back to the wild type receptor OR10. It did not alter the specificity. As you see, the chimeric receptor remains specific to indole. Next, we rescue the segments mid and inner at the same time, so the 2 of them. We mutate all these residues in these 2 segments, and as you see, it changed the specificity, was reverted back to scatol, indicating that the residues here are important. So we went back and looked at only the inner segment, and then we find that also the inner segment revert the specificity. So that's the inner segment that's important here. So we look at the inner segment, and they have only 2 amino acid residues that differ. So let's go work on that. We mutate one at a time. Rescue residue 78, that's to say mutated threonine back to isoleucine, did not influence the specificity. T781I remained specific to indole. Then we mutated only the residue at position 73. Specifically, we revert that position back to the wild type, which contains which revert leucine to alanine 73. And then we found the same specificity. Now we went back then and look at single point mutations. To make certain that we left no stone unturned, we mutate CQ10 M7 from alanine 73 into leucine 73. Sorry, we already knew that this M7 does not participate in the bind pocket, but we have to check everything. So the chimeric receptor was specific to indole, thus corroborated that M7 does not play a role in specificity. It is a single residue at 73 that determines the specificity. We obtained a single point mutation of the wild type receptor. We replace alanine 73 with leucine, specifically CQR10 A73L, behave like CQOR2. That is, the specificity now is to indole with a smaller response to scatol. By contrast, the equivalent mutant in CQR10, position 73 in CQR10, in CQR2 is 74. So in CQR2, 74 is leucine. That's why we did the point mutation before to leucine. Now you mutate that to alanine. With this single point mutation now, we have CQR2L74A behaving like CQA10. That is showing stronger response to scatol and a minor response to indole. In summary, the biochemistry, molecular, and physiology data show unambiguously that the reverse specificity is mediated by a single amino acid residue. By mutating alanine 73 into leucine in CQA10, the receptor behaved like CQA2. By contrast, mutate leucine 74 into alanine, generate a receptor with specificity profile resemble that of OA10. These results were begging for structural biology insights. In the absence of experimental structure of mosquito R and consider that the recent developments of accurate prediction by AlphaFold and Rosetta Fold, we resorted to modeling and docking. We reached out to Vladimir Yarov here at UC Davis and started a collaboration with him and his graduate student Brandon Harris. Vladimir is a go-to person in the UC Davis faculty for protein modeling. Vladimir developed the original Rosetta membrana method for membrana protein structure prediction in collaboration with Dave Baker's group at the University of Washington. Say no more. While Vladimir and Brandon were working on the modeling and docking, we asked whether the single-point mutations would affect the specificity to minor ligands. It's a jargon that we use for compounds activating receptor but eliciting only smaller currents. We call minor ligands sometimes. Both CQA10 and CQA2 responded to phenol, as you see here on the left side of the graph. CQA10 responded to 3,5-dimethylphenol, but CQA2 does not. Now, CQA10 A73L responds to phenol, it's unchanged, but this mutant did not respond to 3,5-dimethylphenol. Contrary to that, when we mutated the CQA2L74A, it responds to 3-dimethylphenol. Taken together, these results suggest that a single-point mutation affects the specificity towards these phenolic minor ligands. We also test the isomers of indole, methyl-indole isomers. In this particular experiment, we left out 3-methyl-indole, scatol, because it elicits such a strong response from the oocytes that could cause desensitization. So we prefer not to make the picture complicated and took it out, and we tested only 1, 2, 4, 5, 6, and 7 methyl indoles. CQA10 showed a preference to 1 and 5 methyl indole. CQA2 did not show a preference for these isomers. The responses were weak to all isomers tested. The single-point mutation ALA73 to leucine recapitulated the CQA response profile, which shows a preference for 1 and 5 methyl indoles. Combined, these results demonstrate that a single amino acid residue, specifically alanine 73, determines the specificity of CQA10 toward the ligands, eliciting either robust or smaller response. Just at the end of last month, the AlphaFold database was expanded to 200 million predicted structures of proteins. So they call it the proteins of the universe. Christian Vincent would be happy to hear about that. Basically, if you have a primary structure, an amino acid sequence of a protein, it's now possible to predict its 3D structure. In this diagram here, the small circle in violet represents the experimental structure, like the structure of Orco, of MHRA, OR5, and the blue circle represents all predicted structures. As of last week, the AlphaFold database includes Culex quinquefaciate protein structures. As you know, this is a neglected species, but I am happy to report that it's now part of the universe. Vladimir generated the new models for the two wild-type receptors, CQR10 and CQR2, and the two single-point mutants, CQR10A73L and CQRA-L74A. Here is the model Rosetta fold structure for CQR10. It's homologous to MHRA-OR5, which is the OR from a primitive species insect reported last year by Delmar Moll and collaborators. Again, Joel Butterwick will talk about this receptor in the presentation in the Frontiers in Chemical Ecology symposium. We have here the overview of the protein. Both AlphaFold and the Rosetta fold are very accurate predictions. Of course, it's better to have an experimental structure, but this is not possible, and I will argue later that in this case the model structure was possibly more helpful than the experimental structure. Here we have AlphaFold in gray, superimposed with Rosetta fold. I compared the AlphaFold prediction structure for CQR10, the one that was just released last week, with the Rosetta fold model generated by Vladimir. There are only minor structural deviation and TM7 as shown here. Now that we had the model structure generated by Vladimir, we want to perform protein ligand docking. First, we validated Rosetta ligand by recapitulating the experimental complex of eugenol with this receptor for a primitive insect, MHRA-OR5. That's the structure reported by Josefina de Marmol. Then we use Rosetta ligand to generate 100,000 docking models for each receptor ligand pair. We have 4 receptors, OR10, OR2, and their single-point mutants, and 2 ligands, catol and indol. Then we generate the docking for each group. We cluster the 10,000 lowest interface energy model, and finally select the 10 large clusters with the lowest interface energy. We therefore generate 10 OR ligand complex model from which we drill our structural hypothesis. Here's one of the poses with the lowest interface energy, highlighting the residues surrounding the ligand. Scatol in green and indol in brown. Sorry that I cannot keep the red and the blue that I have started with the biochemistry and physiology data. Alanine 73 shown here in spacing filling representation. Let's go and see a view from the top. Here's the membrane view of CQR10 on the left and CQR10A73L on the right. Note the proximity of the residue at position 73 leucine in the right side structure. Let's have a closer view of CQR10 scatol bind site. Alanine 73 appears here on the left side. we use the protein-ligand interaction profile to analyze the interaction. The most favorable interaction were non-polar, occasional hydrogen bonding, and occasional parallel pi stacking. This slide highlights the hydrophobic contact and CQR10 scatol model. Of note, 10 out of 10 models shows that the scatol interacts with tyrosine 185. Tyrosine 287 make 8 out of 10 contacts in the models, and so forth. The most striking observation is that there were no contacts between scatol and the alanine 73. Yes, this is the most important residue that determined the specificity, and yet there was no contact. There was no parallel pi stack in this model, and we find 2 hydrogen bonds, hydrogen bond in 2 out of the 10 models. The picture is quite different for CQR10A73L. Again, tyrosine 185 make 10 out of the 10 models, make contact in all of them. The most notorious difference in this model is the residue at 73. In this case here, leucine, that's making contacts with 5 out of the 10 models. We observe hydrogen bonding in 2 models, and the pi stack with tyrosine 185 in 2 models. We made a few conclusions from these models, the most important being that the alanine 73 may indirectly affect the specificity, given that it did not contact the ligand directly. Note that contrary to MHR OR5 eugenol, TM3 does not contribute directly to the binding pocket. It's not surprising, as you know, that in structural biology, one size does not fit all, and also the possibility of multiple binding sites has been suggested before. We surmised that the residues at position 73 modulate the volume of the binding pocket. We align OR10 and OR10-73L by the transmembrane domain 7B, and observe an outward shift of the carbon alpha. Specifically, the carbon in cyan, compared to the one displaying pink, is shifted outwards by about 1 angstrom. Let's have a closer look. The first thought that comes to mind is that the A73L outward shift causes a larger volume of the binding pocket, or there is a constrained space in CQR10 with alanine 73. This would be inconsistent with the physiology data, the oocyte recording. CQR10 with alanine 73 accommodates a larger ligand scatol, and the CQR10A73L with leucine showed a stronger response to a smaller indole. Although carbon alpha is indeed shifted outwards, the carbon delta in leucine represents an overall inward shift of 0.2 to 1.5 angstrom when compared to carbon beta in alanine 73. This inward shift causes a repack of the surrounding side chains, thus reducing the volume of the binding pocket and favor binding to the smaller indole. Indeed, the CQR-A73L indole models show 8 pi stack, whereas CQR10 indole had only 2 of them. So taken together, this data suggested that the binding pocket around the position 73 is tightly regulated. It generates a finely tuned space to accommodate the ligand. The binding pocket is expanded in OR10 with alanine 73, thus better accommodating scatol, whereas the binding pocket in OR10A73L with leucine in that position is contracted best suited for indole. We went back to biochemistry and physiology to test this model-based hypothesis. Mutating OR10 at position 73 to a smaller residue, alanine to glycine. As you know, alanine has 1 methyl group, glycine has no side chain. With this mutate, we saw a further enhancement of the response to scatol. Compared to CQR10, the wild-type receptor, the response of CQR10A73G are stronger, thus suggesting a better fitting of the ligand. Lastly, we tested our hypothesis with a bulk ligand. In the example before, we kept the same ligand, scatol, and mutated the receptor to a smaller residue at 73. Again, glycine has no side chain, and alanine has 1 methyl group as the side chain. Here we test the response of 7 OR variants to 3 ethyl indole. So there is 1 carbon more than scatol. CQOR2 with leucine at that position 74 did not respond to 3 ethyl indole. By contrast, CQOR10, the wild-type receptor with alanine 73, respond in a dose-dependent manner. OR2 mutants with a smaller residue at position 74, namely alanine and glycine, respond to 3 ethyl indole. By contrast, CQR10A73L, again with a bulk residue, did not respond to 3 ethyl indole. However, CQR10A73G, with a smaller residue at position 73, gave a robust response to 3 ethyl indole, even stronger than the wild-type receptor. In conclusion, the accurate modeling doc methods allowed us to put forward hypotheses that can be tested when the high-resolution experimental cryo-EM structure become available. Our major conclusion is that alanine 73 did not form direct contact with ligands, but provide a finely tuned volumetric space to accommodate the oviposition attractant scatol. Please allow me a moment to go back to last century. One of the hallmarks of pheromone-binding proteins discovered in the early 1980s was the 6 cysteine residues. There were no structures, and we solved the puzzle of how the cysteine disulfide bridges are formed by conducting biochemistry and mass spectrometry studies. Soon after we published our work in Feb's Letters, the group of Paolo Pelosi published a similar paper in BBRC. There is an interesting story about these 2 papers, but I'll leave it for another day. Later, John Clark's group, in collaboration with ours, elucidated the first structure of a pheromone-binding protein. You might recall the structure of the pheromone-binding protein from Bombyx mori in complex with Bombicol. At that time, it was confirmed that the structure of the disulfide bridges that we reported earlier were correct. In other words, we advanced the field while waiting for the first structures to be reported. Of course, it would be better to have experimental structure of mosquito R, but you shall not wait until they become available. Lastly, it will be unlikely that one would discover the crucial role that position 73 plays in the specificity of these receptors if you had started from a cryo-EM structure. Would anyone be investigating a residue that does not contact the ligand? It's very unlikely. There are more than 300 amino acid residues in these two proteins that do not contact the ligand. I am glad that we discovered the role played by alanine 73 or leucine 74 in the case of CQ1-2 by a tour de force, and I thank all the collaborators for their effort. We also are very thankful for the continued support from the National Institute of Allergy and Infectious Diseases of the NIH, and I also would like to thank you for your attention, if applicable. Very unusual to be hand clapping on Zoom, right? Thank you very much. So, Alvin, any questions from there?
Yeah. Thank you, Walter, for the wonderful presentation. So from on-site here in Kuala Lumpur, if you guys have any question, yes, Marcelo.
Hi, Walter. Great talk. Thank you. You focused on other than specificity, and you seem to put aside the current changes, the intensity of the current changes. Right. But I was looking at the results you were presenting about position 78, and it seems that there is also a change in intensity of current that is associated with the change in amino acids. When you have a threonine, for example, you seem to have an increased current. Would you discuss about this a little bit? How do you think it will impact the function of the sensory neurons?
Thank you, Marcelo. This is a good question. Thanks very much for sharing the photos that I made, the videos I presented before we started here. Anyhow, so this is a very difficult question because when we deal with the oocytes, there's a variation from oocyte to oocyte, okay? So addressing the issue of sensitivity is not an easy task. So what we focus on here, and the beauty of this model, is that we had two receptors from the same species with two compounds, respond to two compounds in a reverse way, so that we would not have to worry about the oocyte response, because we always have two compounds, and if they reverse the response, then we know that they were attacking the right place. As opposed to many cases that we studied before, that we have one receptor and we want to find the specificity of that receptor, and then sometimes you mutate and you lose entirely the activity. And then that question is, do you lose the activity because the receptor is not properly folded? And what's the problem here? Is really because that site is important or not? So the beauty of this quote-unquote model that we found with OR10 and OR2 is that they both receptors respond to both compounds and in the reverse way. But you're right, like for example, for some reason, every time that we have M5 and M6 mutation there, we note a higher response. So there is something to do with the sensitivity of the receptor. But maybe Joel Butterwick would be in a better position to answer this question because he's the structural biologist here, and he's going to be in the symposium following up that. So there's a question here from, if I may, a question here from the chat that it's from Zain. Do you know the evolutionary history? Is OR2 more ancient than OR10? There is a nice paper now published by you know, that's we found this receptor. You were here in the lab actually when Julia first cloned this receptor, and you got the work going with Chakluedji, and we published the first paper, and then later on the second one that Julian was the first author. Anyhow, so after that, these two receptors, that's something that I didn't have time to elaborate on the presentation, is that these two receptors, they have orthologs in all other species. this is a very interesting. For example, we know that the receptor for DIT, for example, we find in Culex. We cannot find this in Anopheles, for example. But this receptor, WA2 and the WA10, they are in Culex that we found first, then Aedes, Anopheles, and even in Musca domestica now. Jason Pitts reported this work recently, and they wrote a very nice article about the evolutionary history of those receptors. So I would refer to that group for that particular question.
Thank you, Walter. Yeah, is there any other questions from the floor?
People who know me in chemical ecology notice that I made a tremendous effort to speak slowly today because I know that over Zoom is complicated, and I'm knowing chemical ecology to speak a little bit faster than it is, but I tried to pause as much as I could. I hope that there was no problem with the transmission. And also, if I may comment before we have any other question, yesterday there was a comment about the resolution of the video, and this is beyond our control. This is dictated by Zoom. Zoom finds out how many people are watching, and then they reduce the quality of the video to be able to provide to everyone without a problem with the connections. So the quality sometimes is reduced. That's why I knew that when I present that table, no one is going to see that table well, because Zoom is like that anyway. Alvin, anything else from there?
Well, so far there are no questions from the floor over here.
All right, we need to make a very short break anyway to switch the frontiers of in chemical ecology. I'm going to bring all the speakers to the panel and do a little bit of housekeeping work here. So if you do not have any other questions, we shall start the break. What about that, Alvin?
Yeah, sure. Okay. So we'll take a 10 minutes break, Walter?
Yeah, 10 minutes break. We'll start sharply in the hour.
Okay. Thank you very much, Walter.
Thank you. All right, thank you very much for joining us for the Frontiers in Chemical Ecology Symposium, highlighting Rising Star in Chemical Ecology. This is a symposium sponsored by Bedoukian Research Incorporation, BedoukianBio, and that's a new model that we're testing today by having a sample of a meeting of chemical ecology open to the public so that you can democratize chemical ecology and give an opportunity for those who probably would not participate in the chemical ecology. This is a particular special chemical ecology meeting because this is a joint meeting of the International Society of Chemical Ecology and the Asia-Pacific Association of Chemical Ecologists. Nevertheless, Alvin invited me and I agreed to do that, and then we decided that we're going to focus on Rising Star in Chemical Ecology, and then reach out to a sponsor, and the rest of history now. We have the Frontiers in Chemical Ecology Symposium to you today. And without much further ado, let's have a preface for this symposium by Wendell Roelofs, one of our heroes in chemical ecology, one of the pillars of the International Society of Chemical Ecology, a member of the National Academy of Science, recipient of the Wolf Prize, and a very nice fellow. So here's Wendell with his preface directly from Florida.
I want to thank my good friend Walter Leal for getting me involved in this chemical ecology meeting. I'm sorry that I cannot be there in person, especially after seeing the amazing list of award and plenary speakers. I'm honored that Walter asked me to say a few words in preface to this exciting symposium that was organized to highlight the work of assistant professors who are rising stars in the field of chemical ecology. As a world-renowned superstar himself, Walter knows the importance of supporting newcomers in chemical ecology who like to conduct research at the frontiers of this exciting field. Now, just a little bit of history. The International Society of Chemical Ecology was formed in 1983. But I was privileged to be an assistant professor at Cornell University in 1964, where Tom Eisner and Jerry Meinwald were laying the foundation for the field of chemical ecology. They were great mentors for me, and I got to take part in their initial chemical ecology courses and to co-sponsor graduate students. I remember one time after a doctoral exam for Shannon Olson, we had a reception outdoors at a pavilion near a pond. Now, after eating, Tom Eisner went for a stroll around the pond, and when he came back, he said that he had discovered some interesting phenomena in nature for new graduate student projects. I always marveled at his keen sense in discovering new interactions amongst many different organisms and asking critical questions. I was lucky to begin my research back when pheromones were just starting to be decoded, and the whole world was open for the identification of these chemical messengers. I was soon joined in this endeavor by other scientists around the country, notably Jim Tumlinson, who came bursting on the scene in the late 60s with his amazing identification of a complex pheromone blend of the cotton boll weevil. Well, not much was known about pheromones at that time, and so the great thing was that the identification of pheromone structures led to a host of new questions. How did a male moth a mile downwind perceive a pheromone blend with the olfactory hairs on its antenna? And then how did that translate into optomotor anemotactic responses that allowed it to follow the pheromone plume to a calling female? And what hormones controlled the circadian rhythm of female calling and male responding at the same time of day or night? And how does she biosynthesize the unique pheromone compounds? What enzymes are involved in making a specific blend? And what DNA sequences are responsible for producing those enzymes? And finally, how in the world do these mating systems evolve to create new species? The DNA sequences mutate to produce different enzymes that give rise to new chemical structures. But where are the males that respond to these new pheromone compounds? And in order to isolate the new species populations from the mother species? Those are questions from just one chemical messaging system. There is still a plethora of surprises to be discovered by creative scientists who ask and investigate key questions about interactions amongst many organisms at all levels. Stars need to rise up from the new generation of assistant professors who carry out cutting-edge research and push the boundaries of the frontiers of chemical ecology. I wish you all well in this endeavor.
Thank you very much, Wendell. Thank you very much. Wonderful preface, and let's get started. And the first speaker coming from the great state of Vermont. This time I got the state right. So let's have the first presentation by Greg Pask. Greg, the floor is yours.
Thank you so much to Walter Leal for organizing the symposium and using it as a platform to elevate new and emerging scientists in the field. And also big thanks to Alvin He for helping chair this exciting meeting. and especially to the BedoukianBio and Bedoukian for sponsoring the sessions to allow more people around the world to see it. My name is Greg Pask. I'm an assistant professor of biology at Middlebury College in Vermont in the US. I wanted to take this time to share a really cool story that we had when we started to look a lot deeper at a lot of these little tiny microstructures on ant chemosensory cincilla. So many of you have probably, been fascinated by ants in the past, and a lot of times we get very excited when we see ants and how well they work together in teams. And so much of that relies on chemical communication. Ants could be following a trail heading into your kitchen and finding all the food that you left on the ground, or maybe in this example, finding a new place to start a new colony or move the nest. Also, they can use it as a defensive compound. Spraying formic acid at you is a great way to stop you, and also to recruit other ants to start to defend the colony as well. When you have an egg, when you're in the colony, you see all these eggs, you don't know if those eggs were laid by your true queen or by some other invading queen, or maybe a worker who's cheating the system, and they can smell the difference in the eggs. And also, if there's another ant invading the colony, you want to be able to sniff her out. Is that ant that you're interacting with a member of your colony or not? And it all relies on this chemical communication to do so. And a lot of this is reliant on chemicals on the surface of these ants called cuticular hydrocarbons. This is what really transmits a lot of social information in ants, bees, wasps, and termites. Lots of different ants or lots of different insects have these on their cuticle to help them from desiccating and drying out and losing all this moisture that they need. But all these eusocial insects have co-opted these molecules to use them as recognition pheromones for fertility, colony identity, and task distribution. So I like to think of these as little barcodes that each ant is carrying with little identifiers saying, all right, I'm a part of this colony and I'm doing this task within this colony, and then maybe even get down to the individual ant level. Now, chemical ecologists such as ourselves know that these are really a bunch of a big blend of cuticular hydrocarbons at different ratios, and we can oftentimes visualize these as gas chromatograms, chromatograph spectra here that we can look at. And if there are major differences between these, all of a sudden these ants will start to fight with each other. So what do we know about cuticular hydrocarbon detection? This is the Indian jumping ant, one ant that we like to research in our lab. It has been looked at the tip of the antenna, and we have been able to stick electrodes into these little hairs, these little basiconic hairs here, and record information on what they respond to. So you can puff all these different odorants on them, and they will respond and give larger responses. There's a lot of different neurons in these hairs, and we can get this bunch of neuronal activity when we can puff these odors onto them. Not only that, do they respond to cuticular hydrocarbons, they also respond to other odors that are more general odorants that we're more familiar with. My work has taken these olfactory receptor genes out of the ant and stuck them into Drosophila melanogaster, the vinegar fly. And we can take those, we've added one new receptor from this ant, and all of a sudden now it's able to respond to our cuticular hydrocarbon of interest here, in this case, C30. And we can do this across a lot of different members of this family, and we see large responses, mostly in the larger range of the cuticular hydrocarbon options we have here, and they're all specific to certain ones. So from this, we find that these basiconic hairs, which we believe house a lot of these ORs that we're interested in, and these smell receptor genes in particular, respond to all these cuticular hydrocarbons. And this is where we were. This is the work that we've been continuing in my lab at Middlebury. But there's always this really big question of that these molecules are really big. Why are they being smelled in an olfaction context and not just contact with a taste mechanism or contact pheromone detection? So just to compare by size, here's formic acid, which is a common alarm pheromone in ants, compared to that C30 tricontane that I just put up there. This is way bigger. And it's always been a question of how can ants detect such large molecules like these through their antenna? And that's where we were. There's been a lot of questions in this. When ants interact, they mostly will contact each other with their antenna. And a lot of times it's antenna on antenna contact. So there was a nice study here in 2016 where they looked at where ants are touching other ants that they meet with their antenna. And nestmates and non-nestmates, they tend to prefer to touch the antennae and the legs. Specifically for non-nestmates, they tend to look at to contact the antenna of the other ants a lot, meaning that not only are these antenna maybe these like CHC barcode readers, but they are also portraying a lot of these waxy CHC odors out there. But there's also been studies that have looked at how they're able to do this and can it actually be done at a distance. So this is an ant that is on this perch and it can't actually contact what they call a dummy ant here. This ant has been coated in either odor like CHCs from a nestmate or a non-nestmate, and it's able to tell the difference between those without direct contact. So we see this free-moving ant where it can freely interact with it, wants to contact the other ant and question it and probe it with its antenna, but they are able. There must be some sort of limited volatility here. Okay, so this is where the field is at. It's a big question of how do you really reliably detect something that you are also covered in, especially if your antennae are carrying a lot of the signal itself. So a nice thought question, kind of thinking about like, all right, desensitization, but how much do you pick up from other ants? A lot of cool neuromechanisms that have yet to be really elucidated. So we were doing this work with the scanning electron microscope. My student Daphne Haley was doing this with one of our new species they were interested in, the winter firefly. And this was very exciting for us when she started getting really nice imaging from these antennae. We can zoom in a little further and we see this very nice chemosensory hair on the firefly antenna. We see all these little pores along the surface, and we were really, really fascinated by this. But then firefly season comes to an end for us, and we were kind of looking at other things to do. And one of my students who was not involved in this project, but just in the lab, came up with like, maybe we should do some SEM imaging of the ant antenna too. Said this while we were just, chatting at the fly station. I'm like, maybe not. I mean, a lot of it's been done before. We don't really have much to add to the story. This is a good example of why you should not purely listen to a PI's thoughts and feelings, but really start to explore cool questions. So Daphne went outside, found a bunch of these ants, and started to get images of the tip of the antenna, the 10th segment here, and then also zooming in and found these nice big basiconic hairs. And this was immediately very exciting to me because I had seen images like this before, but never detail of these seemingly terminal pores or some sort of depression here, this little microstructure at the distal end of this hair. This is very interesting. So for me and for everybody else in chemical ecology, it was like, all right, olfactory hairs are multiporous, have a lot of pores all along the surface, and they can detect these airborne odors. Insect taste hairs instead have this terminal pore, a single pore, they're uniporous, and this is really what contacts that tastant and allows it to innervate the lymph inside the taste hair. And this seemed to be something right in the middle. So we looked at some more of these and we found this maybe a terminal pore here, some sort of feature at the very distal end, just like our taste hairs, and also a bunch of little pores in the multiporous, more so representing the olfactory hairs here. And we thought, well, maybe, and here's a nice other shot here, all these little pores surrounding by what seems to me to be a larger pore, but we're not quite sure yet, that there was some sort of like hybrid chemosensory hair that could detect both these airborne compounds like a formic acid and like some of the things that are generally more volatile versus something that would be a contact cue like these very large waxy cuticular hydrocarbons that the ants are using for recognition. So we delved further into this. We got nice images. My student Hannah really kind of drove this project forward, getting these beautiful images of these hairs. Here's Camponotus Pennsylvanicus, the eastern carpenter ant. And we see, this is what we've been looking at before. We see these nice kind of depressions at the end, and all these little pores along the outside. So we wanted to look at that along the whole antenna, and when we scanned along all the segments here, we found that they were way more abundant at the distal end of that antenna. We could count 47 on average of these on the distal end, versus you get down to single digits when you get halfway through the antenna. And we can look at this on the dorsal and ventral side of the antenna, and we don't really see much of a bias there, though it looks like it might be trending towards it, but it's not really enough to be significant. So we see that they are there, but they were definitely way more significantly abundant and enriched in their appearance on the distal ends as we go down to the pedestal, which is the base that attaches to the long scape of the antenna, and there's nothing there. So we wanted to look at these. There's a lot of different castes of workers and also reproductives in these ant colonies. Do we see the same differences there? And one of my students, Zach Sieb, started to look at these against all these different castes. He was actually going out, found some queens, actually also found a provider of queens locally in Vermont, about $25 a queen, which was a very nice deal, and a bunch of different ants at different stages in these colonies. And we saw the same general trend, where not really much towards the base of the antenna, and then way more enriched expression of these hairs, a lot of abundance in the distal end, with numbers varying mostly just based on the size of that ant species. So the nanitics are the early workers of these colonies that the first workers that queen lays. They are very small, and then they increase in size as we move along here, with major workers being these ones, having many more of these on the tip of their, these types of hairs on their antennae. So we looked at another ant species, again, the one that we talked about before, this Harpygnathus saltator, the Indian jumping ant, and we were looking to see, could we find similar hairs on the antennal surface? And we did, and we started to isolate these basiconic hairs that have been characterized before by Jürgen Liebig's lab. And lo and behold, we looked closer and we found this little depression there. We zoom in further and we see that little depression, and we see a bunch of these small little pores along the surface. Again, this unusual combination of multiporous with maybe something uniporous, or even just a decrease, or like a little depression in that surface. Did the same thing. They are also highly abundant on the distal end of the antenna. And we don't really see a major dorsal ventral difference here, which for us makes sense because ants are contacting other ants with all 360 degrees area of their antennae. And when we look at kind of the older images that have been taken from these, we really see maybe what we think is the difference between why these weren't able to be illuminated before. If you squint close, you can see little remnants of maybe a little groove or depression in the ends of these hairs. Here's another one here. And we think the major difference is that we were using much lower accelerating voltages of our electron beam to really provide more superficial detail, to look at just the surface of those of these hairs, as opposed to kind of getting deeper into the tissue and maybe missing some of the features. So this was accelerated at about 20 kilovolts, and this is about 5 kilovolts. And we're able to see the differences at this level and get the images that we saw back here to resolve all these different pores. So we started to kind of look at other ants. We had several ants. This is the Allegheny mound ant right in our backyard. We're able to check these out, and this is in the Formica genus, and we're able to see a very similar structure. Okay, let's move outside of our carpenter ant realm, and we moved into harvester ants. This is the Pogonomyrmex occidentalis, and we see maybe this is where we started to think maybe this isn't necessarily a pore, or at least in all ants, but maybe something more of like this little depression. We see a bunch of these small pores on the surface, and then this, maybe that's open, maybe it's closed, maybe it's open and clogged in these images, but we're saying this is kind of more of a depression that might increase the sensory surface area at this point. Then we looked at leafcutter ants, and we didn't see it at all. And this was really interesting to us. And these were big, large, super major ants, so it wasn't necessarily a size thing. And we saw a bunch of little kind of ridges and textures here, which also can increase surface area, but we don't see any of these terminal depressions at the end of these hairs. All right, so let's move into some other ants. We moved into Argentine ants and odorous house ants and clonal raider ants, and we don't find these at all as well. We might see some like nice grooving here. We can see this very much here on this clonal raider ant, which again increases the surface area, which you would want to have a larger sensory surface for a contact hair, but we don't see these. So as we're kind of expanding out into other insects, or other ants, we're not finding these as much. So what can we understand this through the context of these ant subfamilies and how they've evolved? And these are all the ants that have been, the ant subfamilies that have been characterized here. We tried to go get ants that we just showed from the larger families of, larger subfamilies of ants. If we look at them, we find some very confusing findings in which the green here is finding some sort of depression in the tip of the hair, and the red is no depression. And within the Myrmicinae, we find a depression on the harvester ant and not on the leafcutter ant. In the Formicinae, we see both the eastern carpenter ant and the Allegheny mound ant have it. When we move into the Argentine ants and the odorous house ants, nothing. The clonal raider ant, nothing. But then if we move further out to the Poneran ants with those our Harpydanthus saltus, where we see it. So we're looking at this and trying to understand, we see no real clear evolutionary pattern, and there could be multiple steps of loss of this microstructure or gains, and we're just really not sure at this point. So, but what we do know is, and what we can kind of wrap our heads around, is that we have these ants that interact. And when ants will interact with each other, they're oftentimes contacting them and contacting each other with their antennae. And this makes sense because that large terminal depression and the abundance at the very end of the antenna really aligns with what one would assume would be a contact model for how they're sensing this. If they were just picking this up from airborne signals, it wouldn't necessarily need to be so abundant at the tip of the antenna. Also, these basiconic hairs have been recorded from using electrophysiology. They respond to these hydrocarbons that have limited volatility. We can artificially boost the volatility with a heated puff or coming out of a GC, but they also respond to the smaller airborne odorants. Perhaps the large terminal depression is responsible for the direct contact with the CHCs, and maybe the small hairs are responsible for kind of the detection of the smaller odorants. And then these hairs are very important for nestmate recognition and a lot of the social behaviors. These are not found. You cannot find these at all in male ants, which we tend to think about as like flying packets of sperm that are there to just mate with queens and sometimes workers. And the males don't really rely on the CHC detection at all, or that has, you know, they can sense it in other hairs, but not really, they don't need to use it as much for their lifestyles in the colony or without the colony. So we think that this might be this kind of potential multi-purpose chemosensory hair is just another one of these great model, like examples of structure determining function. So we have a lot of this information about the chemistry of CHCs and the sensory function of them. We have a lot of behavioral data of just free moving ants and how they interact with contact. And there are all these questions. And we think that kind of this might be a nice model and understanding of like, all right, there's this terminal depression at the end of the hair. That might be what we're looking for here. And again, I used to present data on this and images of this based on Drosophila hairs and exploring kind of beyond model insects. Maybe these model insects aren't models for the rest of the million plus insects that we know are out there. So there's way more to be done that we necessarily can do, some we can't do. A lot more ant species to explore to see if we can find this microstructure around them. It was very cool when we put this up on bioRxiv recently that Santiago Ramirez of UC Davis said, oh yeah, back in 2008, we found similar structures and we just weren't able to resolve them enough. So the fact that other people are finding these is kind of fun to kind of fit with prior knowledge and, prior time spent at the SEM. We also, I am mostly a functional olfaction person. We're looking at a lot of receptor function and electrophysiology. We've thought about a lot, could we try to find a way to determine, is that just a depression and a decreased well of surface area on the end? Or is it actually a true pore like a taste sensillum? And we don't think we'll be able to do this because the thought was all right, we could get a little pipette tip and cover that pore and kind of do the typical taste sensilla preparation. But we also have all those tiny little pores around that odorants could be moving through as well. So we wouldn't be able to resolve what we're hoping to pick up there. So what we think it really needs to be done is using some transmission electron microscopy, or TEM, to kind of study this difference, get some kind of slices through that, and see if we can determine that, is it a pore or maybe just a depression or a pore in some species. And this is technology that we can't do. There's a lot of great TEM studies of different sensory hairs and insects, and hopefully others that are equipped to do this can pull this off and start kind of pushing the field forward and kind of building off of the imaging that we found. So with that, I'd like to thank everybody that was involved in this work. Hannah Gellert really drove this project forward after Daphne started to get the work done there, and Zach stepped in and got us a lot of additional cast-specific data for Camponotus. Other Pask lab members for helping collect ants and general support and looking over this manuscript. Jody Smith was really instrumental in helping us use the SEM and get started on that. And that SEM was provided by the Middlebury Geology Department. I'd like to thank a lot of individuals for providing us ants that we did not have from across the country, and also Alex Weil for providing, as to the whole field, all these great images of ants and other insects that we were able to use. So with that, thank you, and I'm happy to take some questions.
Thank you, Greg. That's a wonderful presentation. In the interest of time, let's move quickly here. There's a question from someone anonymous. Why more basiconica sensilla at the distal end? Does this in any way relate to their role?
Yeah, so that's a great question. It's one that we've been thinking about a lot. We really think that it aligns with the behaviors that we see, that they are probing each other with their antennas, primarily the tips of their antennae. So we think that might align with that contact model. That there's still some debate in the field about how ants are doing this, but what we see when we watch ants do this is a lot of contact.
Alvin, any question from the podium there?
So far, none. Yeah, there is one. There is two. Okay.
Okay. So let's get the question from there.
Yes. Dr. Watanabe, can you please go to the mic, please? Thank you.
Hi, thanks for a great talk. My question is related to the one from the chat. The ant species where you didn't find these peculiar depression structures, what do we know of their behaviors? Do they maybe differ from the ones where you found them? That they antennate differently, or maybe more the abdomen or other body structures?
That's an excellent question. When we look, the species that we had in-house are primarily the two that we have, and we've looked at a lot of these behavioral observations, but it's my understanding that a lot of these ants, whether it's Argentine ants or the clonal raider ants, at least human observation, real time, that they are doing these same kinds of probing behaviors, and tend to always kind of meet at the front, whether it's nestmates and maybe they probe each other, and then there's a trophylactic response where they're starting to share food, or if maybe they find out that they are not nestmates, and then all of a sudden they start to wrestle and get aggressive towards each other. So these occur, and these kinds of behaviors occur, and we think that they are still doing this heavy dose of contact, but it's also species that we are just receiving from others, and when we can get them in-house, we could kind of start to explore these, hopefully with like high-speed camera interactions.
We might have
Thank you.
to move on and take some questions for later, but I'm going to mention here two questions very briefly, if you can combine them and move forward. Did you look at the work of Mamiko Zak? She showed the terminal pore and wall pores on antenna sensilla some years ago. And another one is that very interesting talk. Have you looked at for those hairs on any bee species?
So yes, Mamiko has shown some of these pores in a recent ant learning paper with Camponotus japonicus. There's been some variation of some publications from that exact species have shown a pore, others have not. It might be about the resolution and the accelerating voltage of the SEM. So we're trying to just show that, hey, there's more in a lot of these different species out there. And yeah, when we look at old papers, we can kind of find that description, and we can like kind of squint and look at the images and see, oh yeah, there's the remnants, but it's much lower resolution and clarity that we'd like. In bees, we've looked at bees and wasps. We've looked at the honeybee and bumblebee in the US, and also several species of wasps, and we do not find these structures.
All right, thanks very much, Greg. Maybe we'll come back to that at the end, but now we have to move to TIC, Chemical Ecology, as well as send the virtual podium to Nova Scotia, where Nicoletta Faraone is doing what Jeremiah Wild would call chemical prospecting. Here's Nicoletta.
Hello, everybody. My name is Nicoletta Faraone, and I'm an assistant professor in the chemistry department at Acadia University, Wolf in Nova Scotia, Canada. And today I'm going to talk to you about host detection by ticks and effect of repellent exposure. But before to start, I would like to thank all the members of the Faraone Lab, my students, research assistants, and postdocs that worked so hard and with such an incredible enthusiasm on their project. Also special thanks to the funding agency that over the years supported my research and my collaborators at Acadia University and other academic and research institutions. So we're all experiencing climate change with a steady increase in temperature, and these environmental conditions are promoting the rising of vector-borne diseases, such as Lyme disease. Now, Lyme disease is a major vector-borne bacterial disease in Canada and United States. It is caused by a pathogen, Borrelia burgdorferi, and transmitted among hosts and humans primarily by the black leg tick, Ixodes scapularis. So this tick species spread the disease in the northern, mid-Atlantic, and north-central United States. You can see in this area. Now, risk area are all around the northern hemisphere, and as you can see, the number of cases in Canada has grown exponentially in just 10 years. Now, Nova Scotia, we're located here, represent a high-risk location. So this is particularly, this situation is particularly serious in the Atlantic provinces, and Nova Scotia is considered a hotspot for tick population and abundance, and it represents the Canadian province with the highest risk in contracting Lyme disease from a tick bite. As you can see here from the map, these are all the counties on Nova Scotia, and the dark blue represent the zone where there is a higher risk of contracting Lyme. So you can see whole Nova Scotia is basically dark blue. Climate change models predict that Nova Scotia is very close to having a suitable climate for tick establishment across the country. Now, the threat of disease transmission has led to an increased demand for effective and environmentally friendly tick repellent products. And a good example is offered by natural products and essential oils, which are very well known for their valuable properties as a repellent and safe to use active ingredient. But finding a suitable product as an arthropod repellent may represent a challenge, in part because of the lack of knowledge of the tick chemiosensory system. So how does it actually work? Until recently, very little research has been done to understand how ticks detect repellents and whether ticks can discriminate between repellency due to olfaction versus that from contact chemioreceptions. It is generally recognized that ticks detect host cues using sensilla located on the tarsi of the front legs. And these sensilla are organized in a very particular organ represented here, Haller's organs. And recently, we have reported that pedipalps have been described as multimodal organs that is involved in both olfactory and taste detection. Now, ticks rely very much on chemiosensation to identify and locate hosts for a successful blood meal because they're almost totally blind. Now, another important factor that requires attention is whether bacterial infection might change the behavior or the sensitivity in the ability of ticks to respond to other stimuli. In fact, it has been observed that Ixodes ticks infected by Borrelia burgdorferi survive better under unfavorable conditions of temperature and humidity, different level of energy reserves, and they have shown increased activity in questing behavior. So based on these very interesting questions, we wanted to investigate the tick olfaction in order to better understand how volatile organic compounds are detected, and then use this information to develop new repellent products and innovative methods for the prevention of tick-borne diseases. We studied the neurophysiology of ticks olfaction in the context of behaviorally relevant volatile stimuli, and also we investigated how these volatiles may alter the electrophysiological and behavioral response of ticks to other stimuli. Ultimately, all this information will be used to develop novel repellents and caricidal products. Now, the question that we want to answer, so we want to know, does the presence of a repellent impact the ability of ticks to detect attractants? And has the repellent exposure a long-lasting inhibitory effect on the ability of ticks to detect attractant and potential host? And does the pathogen impact the ability, the tick ability to respond to attractant and or repellent? So to answer to this question, we designed a specific bioassay that would help us to tackle down all these unknowns. So what happens if the tick is exposed to a repellent for a prolonged time? Because we know that ticks, when they are exposed, for example, in bioassay, they are repelled right away. But what if this exposure is prolonged? So we measure the electrophysiological response of ticks to butyric acid before and after exposure to repellents. And for repellents, we selected lemongrass, essential oil, and DEET. Now here in the table, I'll guide you what I have reported. So this is the electrophysiological response of blackleg ticks before and after the exposure. So here we have the treatment pre-exposure, and we have recorded the response of the tick to control, which is just hexane, to DEET, lemongrass, and butyric acid. And as you can see, the only compound that elicits a significant electrophysiological response is butyric acid. Then we have exposed the ticks to this repellent product. So in the post-exposure assay, we have exposed the tick for 20 minutes to a repellent, lemongrass or DEET, using a fumigation chamber here represented. And then we have recorded the electrophysiological response towards butyric acid. And as you can see here, the exposure to hexane and DEET does not really change the ability of the tick to detect butyric acid. However, after the exposure to lemongrass, the long-term exposure, this significantly reduced the tick response, and there is a decrease in the action potential here. Then we wanted to see if this repellent exposure would impact ticks' behavior. So we perform a behavioral assay using a Y-tube assay, a Y-tube setup to assess the tick behavior after the exposure. And we tested the behavioral response, so the attraction toward butyric acid, right after the fumigation or after 24 hours the exposure, the fumigation exposure. And what we found was quite interesting. So as you can see, prior to the exposure, ticks tend to be attracted to butyric acid. And this trend is conserved after the exposure to hexane, which is our control, and after the exposure to DEET. However, they seem to be repelled actually by butyric acid, and instead they go on the other side after the exposure to lemongrass. Now, after 24 hours from the lemongrass exposure, the situation seems to change again, and now ticks are again attracted by butyric acid. However, this we need to take into account that 50% of the tested ticks after 24 hours fumigation exposure did not respond. So it's hard to draw any conclusion here. Here we have a table that reported the percentage of responding and non-responding ticks in Y-tube bioassay, and you can see that the post-exposure we have basically around around the 90% ticks of that responded to the stimuli, to butyric acid, but here after 24 hours lots of ticks did not respond. So since we see such a decline in tick response, we then wanted to see if the exposure to the treatment would have had any long-term impact on the tick chemiosensory system and on the morphology of the tick chemiosensory system. So we looked specifically at the Haller's organs, but no significant changes or damages were detectable on the sensilla. So these results were very interesting, but consider that they've been done on lab-reared ticks. So we wanted to see basically if the presence of the bacteria, so the bacterial infection, would have an impact on ticks' ability to detect attractant and respond to repellent. So we have been collecting ticks in the field, and we are performing the same type of experiment on wild ticks, so doing electrophysiology and behavioral assay, and we are doing this not just on Ixodes scapularis, but also on Dermacentor variabilis. Now, we have collected wild ticks that are being using in this experiment, and after they've been stored for further analysis because we are going to analyze the bacterial count, the level of infection, the type of infection. So from the electrophysiological response of adult females of Ixodes scapularis being collected in the wild, again, same idea, exposed to different treatment and test for response towards butyric acid. As you can see here, we have a similar trend, but also we have a decrease of the response towards butyric acid after the exposure to DEET. So DEET and lemongrass seems to have an impact on the tick ability to detect butyric acid after fumigation exposure. And currently we are performing behavioral assay on wild ticks, on Ixodes scapularis. Then we want also to see if there is any differences in a response between sexes. So we've been testing Dermacentor variabilis, so the American dog ticks, and we wanted to see the response of males versus females. And here we have recorded the response we recorded the response towards the butyric acid after the exposure to hexane or to lemongrass. And you can see that here also we have response to, we have a decline in the response, and the behavior reported, it's the same between females and males. So in summary, we have found that the ability of ticks to detect attractants delivered together with repellents is significantly reduced. So is there any masking effect that we are seeing? Is there any impact on the at the sensilla level that is justifying why the exposure of this repellent compound impact the detection of attractant in ticks? So it's something that we need to still clarify. The exposure to repellents such as lemongrass inhibited the capacity of ticks to detect attractant afterwards, and the exposure to treatment reduced the activity of ticks after 24 hours. Now, wild ticks seem to respond similarly to lab rear ticks, and but we need to correlate this information with the infection level and bacterial count to see if there is any impact of the chemiosensory system of ticks and how they detect volatiles. So other activities and other projects that that I'm involved with that I want to tell you about is the Canadian Lyme Disease Research Network. So there is a very strong need to build the research capacity to address the emerging threat of Lyme disease in Canada. So we have established the Canadian Lyme Disease Research Network to improve prevention, diagnosis, and treatment of Lyme disease in Canada. So we've been collecting and doing surveillance in the province, and all those ticks will be tested. So the scapularis will be tested for Lyme disease to have a better mapping of the Lyme disease distribution. Also, there is a very cool app called the eTick, and is a public platform that's been developed by Canadian universities, and Acadia University, and Dr. Kirk Hillier is directly involved in this. And this is an image-based identification and population monitoring of ticks in Canada, and provides up-to-date information about what tick species are currently being found, and as well as changes in their geographical distribution. And since 2017, you can see that more, almost 31,000 species, 31,000 pictures have been submitted for the identification. So this app is very cool because provided a live maps of ticks distribution all around Canada. Also, another very cool project that we're working on it looks at the questing activity and preference by host tick nymphs and adults in different vegetation and habitats. So this project is led by Dr. Sarah Corte and is funded through CanLyme, which is the Canadian Association of Lyme Disease. Now, what we want to know here is whether ticks prefer certain plant species and habitats for questing, and if this preference is associated to host species, and finally, if this association host habitat is regulated by specific semiochemicals. So it's a lot of cool activity, and that hopefully will help us to understand better how ticks respond to host and volatile organic compound, and how this information can be used to develop proper repellent product that would protect us against the transmission of diseases. Thank you very much, and I'll take any questions.
Thank you, Nicoletta. Very nice presentation. We have some questions here. If people want to ask questions, please move to the mic there so that we save time. Danielle says, quite interesting. So how do you account for the non-responsive ticks to the Y-tube bioassay exposure?
So thank you for the question. What probably we know that lemongrass and also DEET and hexane might have some acariciidal effect. So I think the non-responding would have impact somehow the physiology of the ticks. That's why they didn't respond. Now we are repeating the experiment using different concentration, and we're trying to see if this 24 hours, after 24 hours, ticks don't recover, so there is an actual damage. And ideally, it will be also cool to see if the exposure will impact the internal morphology. So looking at, for example, the synganglion, or looking at more specifically the sensilla, because we took just SCM picture, but there is so much more to see if to understand, you know, what the fumigation does to the ticks.
Very good. And Alvin, if there's no question from there, I'll keep with the chat from here. Cameron Schlager is asking, is the longevity of the tick exposed to lemongrass or DEET shortened by the exposure, or is it the interaction of these repellents with the ticks only an odor receptor process?
Yeah, I mean, that's why, because we are also concurrently performing acaricidal tests using these essential oils, and we know that lemongrass has an acaricidal effect. However, this question, this observation is really interesting, because we are trying to see the mode of action of the fumigation, and this is the long-term effect. So it might be that it's just impacting the chemiosensory system, so the tick is unable to detect anything and then move, basically. Because if it doesn't detect any volatiles and any attractant or repellent, the ticks won't be motivated to move and climb the white tube.
Alvin, you can interrupt at any time, okay? If you have a question there. Yeah, yes, go ahead.
Sure, no problem. I'm monitoring from here.
No question from there, right? Is there a question from there? Just interrupt at any time. Aditya is asking, is the relatively quick increase in tick population correlate with increase or invasion of some other plant animal species?
This is a very good question. What we see, especially in Canada, is I would say the increment of deers that are invading all the area that are density with high density population. So of course the expansion of the reservoir of ticks, it's promoting the expansion and the spread of the ticks. So it's a very, very good correlation. And we don't have to forget it's also climate change that is helping, is creating a warmer, and we have a warmer winter. So there is a very promoting the best environment for ticks to thrive.
David, thanks for your presentation. So do we? Tick in nature can be infected by multiple pathogens. Can this affect response of wild ticks to your compounds?
Absolutely. So we're going to look at not just Borrelia burgdorferi, but there are other bacteria that we're going to look at it. So we're going to do multiple analysis and looking at bacteria, virus, and other pathogens. So of course, the main idea was Borrelia because it's very common in Nova Scotia, and there is a high percentage of ixodiscapularis that are infected with Borrelia, but we think there will be the synergistic effect of the bacteria that somehow impact the ticks, how they respond to a volatile, attractant or repellent.
Aditi, thanks you for answering her question. What time do you have in Nova Scotia now? What time?
It's 11 p.m.
11 p.m. You stay late until then. I hope you stay. Okay. I hope you continue with us so that there will be more questions by the end. So let's move on. Let's move on. Then. Thank you. Thanks very much. Let's move on now. We send the podium now, the virtual podium, to State College, to Penn State, where one of our heroes, Jim Tomlinson, was home to Jim Tomlinson for 2 decades. Penn State is one of the centers of chemical ecology in the United States. And there, Erika Machtinger is study also ticks, and she's putting ecology in the chemical ecology, so gives more emphasis to the ecology side of the work. So let's then send the podium now to Erika Machtinger and attending her presentation, remote presentation. Erika, the floor is yours.
Hello, everyone. I am really honored to be included in this awesome group of speakers and want to thank Dr. Leal again for the invitation and all of his hard work organizing and promoting this symposium. So today I'll be discussing some behavioral ecology leading into chemical ecology work with the black-legged tick, which is an important vector here in North America. So I want to make sure I have time to acknowledge the people who have worked really hard on this project, including Arash Maliki, who is a student of Jim Tumlinson's and now a research tech in my lab. I want to thank Jim again. I couldn't thank him enough while he was with us for his guidance, pushing us to look closer at tick-host relationships, as well as Karen Poe and Jessica Brown, who have worked on different aspects of this project. And I'd also like to thank our USDA and NIH funders for the support to conduct this work. So I do like to shape these talks into stories. So let's talk about the villain in our story, which is the black-legged tick. Now for our story, this tick is our villain, but I do want to add a footnote that ticks and parasites play many important ecological roles, like being food for wildlife and regulating some populations. So while this tick is our villain in terms of public health, it is not a villain in the ecological sense. But this is a very common tick in the United States that continues to increase its range. And this tick in particular is of medical and veterinary concern because it is the vector of the pathogen that causes Lyme disease, which in the United States is the most common vector-borne disease. So I'm going to focus on the state of Pennsylvania, which is where I currently live, to demonstrate the current state of Lyme disease. So basically the bottom line is that it's not good. Not only is Lyme disease the most common vector-borne disease, but all cases across the country continue to rise, which you can see here in this figure, with nearly double the number of 2010 cases from around 2015 and on. Now keep in mind, this is only one state out of the country that is represented here, and that total cases are estimated to be about 10 times what is confirmed by diagnostic tests. But here's the problem. So one of the challenges we face in control of these ticks is that there really isn't that much information on tick ecology, especially when you compare it to other important vectors like mosquitoes. So one of the focuses of my lab is tick-host relationships and tick control. And it was during one of these studies on tick-host species composition in Northeast forests that we observed a really interesting phenomenon with two sympatric animals, white-footed mice, which you can see in the image on the left, and redback voles, which are on the right. These two species not only were trapped in the same environments, but overall are pretty similar in morphometric measurements like body length and weight. But over a 2-year trapping period, we saw extreme differences in the distribution of ticks between these two species. So we found that not only were the mice infested with ticks more often, they were also more occurrences of individual mice having high numbers of ticks, like 25 or more ticks, whereas the highest ever found on a vole was 5, and that only occurred once in 2 years. So the average number of ticks found on a white-footed mouse was just over 4, and it was less than half a tick per vole overall. Now again, these animals were roughly the same size, weight, trapped in the same areas. So the obvious question here is why were we seeing these differences? So let's bookmark that thought for just a moment, because before we talk about these differences in attachment by host species, I want to give a little bit of background about tick hosts and why the white-footed mouse is so important in this story. So black-legged ticks take 3 blood meals from 3 different hosts throughout their life cycle. So smaller animals like rodents are the primary hosts for the juvenile stages, and adult ticks tend to feed on large animals like deer. Now the little white-footed mouse is so important to this story because it's a really important reservoir for the Lyme disease pathogen. It's estimated that about 90% of ticks that feed on an infected mouse will become infected with the Lyme disease pathogen. However, even though a relatively recent study by Halsey in 2018 found that nearly 80% of black-legged tick studies focus on mice and deer, black-legged ticks have been considered generalists throughout their life cycle. So then the question is, if ticks are generalists, why are so many more feeding on white-footed mice? So that was the central question to what led us into this next series of studies. And because so little is known about the ecology of ticks, including chemical ecology, we started with behavior. So animal behavior is driven by many environmental cues like auditory, gustatory, visual cues, tactile cues, and of course, chemical cues. And often talks in the realm of chemical ecology have a lot of chemistry, but looking for chemicals first without establishing a suitable bioassay to evaluate behavior is like looking for a white cat in a snowstorm. So in other words, it's very challenging. So what we've been trying to focus on is the behavior to understand these tick-host relationships. So what do we already know about black-legged tick chemical ecology? There are a lot of really fragmented pieces of information. We know that ticks demonstrate distinct heterogeneity within species, with about 20% of animals carrying 80% of the total tick burden. There are many theories to why this is, but studies have yet to find a consistent common link to the causality. We know that Sonnenschein and his colleagues in 2003 found that nymphal black-legged ticks were arrested by specific compounds from conspecific ticks. And recently, Josek and their collaborators found that adult ticks demonstrated electrophysiological activity when exposed to specific crucials, but behavioral responses were disappointing in the lab. We have yet to see any evaluation of black-legged tick attraction to live hosts. Now, fortunately or unfortunately, this is a big question to tackle because where do you start? Is tick attraction to hosts based on tick behavior, or are they truly generalists and successful attachment is in response to host behavior or physiological status? Or, more likely, is it a combination of many factors that drive tick attachment and successful feeding? So we decided to start with the most simplistic questions. 1, are ticks attracted to hosts? And 2, how do hosts respond to ticks? So our main objectives here were to look at black-legged tick responses to white-footed mice, mice with tick infestations, tick responses to meadow voles, and then no-choice assays comparing mice and voles to get to that question why we were seeing such big differences in the field with tick attachment. And you may be asking why with and without tick infestations. So we thought back to the Sonnenschein study and thought that it would make sense if ticks could pick up on the pheromones of conspecific ticks already feeding and take that as a cue that they could also be successful as well, perhaps leading to aggregation on a single host animal. So our bioassay setup looked like this for the most part. We've now placed the jars vertically to prevent our rodent subjects from eating the lids. We have custom jars to house our animal hosts. These are connected to 2 of the 4 ports on our 4-port olfactometer. Now, all of these trials were conducted with the appropriate animal welfare approvals. So during these trials, temperature and humidity monitors are placed within the jars. We evaluate each side for CO2 output. And for each replicate, we use 10 pathogen-free black-legged ticks from colony. And these are placed in the 4-port. Now, we had several iterations of this setup that we tried before finally landing on this. Our experience with Y-tubes and U-tubes, which are often methods used for olfaction-related behavior, showed that black-legged tick nymphs did not move in some assays, regardless of orientation of the Y-tube, and it was hard to infer if this was due to a repellent effect of the odor or experimental setup. So we decided to use the 4-port, which provides us a little bit of flexibility in choices as well. Now this is a top-down view of our olfactometer where you can see our determination of a choice line on the left side and the right figure examples of nymphs that have made a choice in one of the odor ports. So we set out to evaluate tick attraction to mice, thinking that ticks would have some degree of attraction to a common host and that would see perhaps less attraction to voles, which would explain what we were seeing in the field. But what we got was a major story plot twist. Basically, we didn't see this coming. Ticks don't choose mice. Over and over again, black-legged tick nymphs were actively repelled, which means they walked away from the odor of live mice, which was a natural host, and towards the clean air. So overall, nearly 3/4 of our tested ticks chose clean air over the mouse odor or the mouse host. So, okay, maybe it is something like tick infestation that gives ticks a cue that biting may be more successful. If ticks before you are feeding successfully, maybe you can too. However, when we looked at infested mice against a clean air control, we saw fewer ticks responding to the mouse host, so about 85% of our ticks choosing the clean air control. We did a no-choice assay, and when black-legged ticks were put in a no-choice situation, they selected rodents with ticks and rodents without ticks equally, suggesting that there were no differences between these treatments in tick attraction. So then I got thinking, what if there was a confounding variable in our setup? So if you look closely at this image, you can see a mouse in our jar, but also quite a bit of fecal matter and urine. And we know that odors that are commonly found in feces like guanine elicit an arrestment response from ticks. So what if there were some repellent compounds or arrestment compounds in the feces that made ticks stop that was influencing our results? But how do you tell a mouse not to defecate or urinate? So what we did was anesthetize them so they wouldn't produce any waste. We knocked them out for 45 minutes and conducted additional trials, and what we found was that the repellency was actually more likely. So 93% of our ticks were repelled by the mouse host when it was anesthetized without any of those waste products. Okay, so that was not what we were expecting. But what about the voles? So trials were run looking at vole odors versus clean air, and nearly 70% of ticks were repelled by the vole odors. This was very similar to our mouse response, which is about 74%. So when we looked at voles compared to mice, there were slightly more ticks, just in basic percent response that moved towards the mice, but these results were not significant. So this wasn't at all what we were expecting to happen, of course, and goes against the most basic thought exercise. So why would a parasite be repelled by its host? So we started brainstorming and coming up with alternative reasons and experiments that we needed to do to start investigating this a bit deeper. So the first is maybe attraction to hosts is more close range. Maybe they have to be closer to them. Or maybe low attraction is due to niche partitioning. Or perhaps individual hosts instead of a population of hosts have more influence over tick attraction. Or maybe we're looking at the wrong behavior. For insects, attraction is active movement towards the attractant, but maybe for ticks, attraction is a little bit different. Or maybe attachment really is totally random and there is no innate attraction, and biting success is more host influenced. So I'm going to go through these a little bit deeper to give you all a little bit of a sneak peek about what we're doing next. So the first thought we had is maybe attraction is low due to niche partitioning. And back in 2008, Brisson found that the distribution of larval ticks on white-footed mouse hosts was actually really low, like only 10 to 15% of the total larval tick population fed on mice, whereas the remaining ticks were found on other hosts. So nymphal attraction in our assay was about 25%, so perhaps these ticks have some differential attraction that allows them to separate over a range of hosts and not overwhelm any one species, which would be bad for them as a parasite. Another thought we had was perhaps attractive cues are given off at the individual level and not at the population level. So if you take a look at the bar chart on the right, you can see that in 2011, Bennell found that nearly 40% of animals that were considered sick were infested with ticks, whereas healthy animals did not have that parasite burden. So ticks need to override the immune response of their host in order to feed successfully, and immune function can influence metabolites, which could be read by ticks as being more or less risky targets. And the animals that we used were from our established colony that are healthy, well-fed, low stress. So perhaps we aren't seeing attraction because our animals aren't at the right physiological state. And we intend to test this hypothesis with induced short-term immunity function triggers in our mice that will allow us to evaluate tick responses to basically sick animals. We also thought, well, ticks aren't insects. We are approaching this from a very insect perspective, but what if attraction to ticks means something different? So if you think about it, these ticks are not very mobile. They sit and wait. It wouldn't make a lot of biological sense to walk towards a host. By the time you get there, that host would have left the area. However, it would make sense to have these odors induce a different behavior, like climbing or questing. So we have a setup to start evaluating these behaviors that allows for climbing and to record questing behavior over the duration of the trial. So finally, there is a chance that some component of tick host selection is just random and that ticks only respond to other sensory cues like heat and vibration in concert with CO2. We are tackling this component of the tick host selection process as well in my lab. So a student in ecology in my lab, Jessica Brown, is using EthoVision software to evaluate rodent behavior after tick attachment and is comparing different levels of tick attachment among species of rodents, including mice and voles. And you can see in our setup here with the phenotyper cages that house the rodents on the left, and on the right is what the image looks like with each of those yellow blobs, tracing a rodent that's in the cage and their behaviors, including grooming behavior. So to wrap up here, we didn't get into any of the chemistry or chemicals or anything other than strictly behavior related to tick host attraction, but all of this behavior work is setting that foundation for us to really understand the process of how ticks go from host seeking to actually on a host, and whether that's a selective process or a random one. We will continue to explore alternative tick behaviors which may indicate attraction, continue to work with the host to evaluate their role in successful feeding, and we will continue to evaluate the compounds associated with the hosts for their role in any of the behaviors we've identified in the bioassay, including repellency. So I want to thank everyone again for taking the time to join us and looking forward to answering any questions you may have.
Thank you very much, Erika. Alvin, are there questions from there?
Yes, there is. Yes, there is. Renee, can you go to the mic? Thank you. Renee has a question.
Hi, can you hear me?
Yes, you can hear you well.
Okay. So I had a question about the alternative cues that you were talking about. It struck me that the shrews had the highest tick load when you did, you showed some older data.
Right. Yes.
And the shrews were had a higher load, and shrews have a much higher metabolic rate compared to other mice. So I was wondering if you could compare to the vole and the mice, the white-footed mice, if you compare their metabolic rates, and I'm sure perhaps even CO2 might be, if you have a higher metabolic rate, then you would have higher CO2. So that might be something that, I'm sure you're already looking at. But maybe the shrews will give you the clue as to how to proceed with that investigation. Lovely work. Thank you.
That's an excellent comment. We have been measuring CO2. Actually, our voles have higher CO2 than our mice, but they are the ones with fewer ticks. But you're right that shrews are notoriously high. Their metabolic rates are notoriously high. They're just a little bit more challenging to keep alive. So we haven't tried that yet, but it is an excellent thought, and working with somebody who has some of the equipment to look at that is an excellent thought.
You raised so many questions, Erika, that if people will get out of questions. But there is one here on the chat from Zain Said asking, were the overall numbers different, not percent, when both animals, same or different, were present as compared to only one treatment versus control?
So overall numbers, yeah, when we have equal numbers of trials, the numbers are very similar, but I will add that we have run into the same issue that Nicoletta has run into, and I think Zane has run into as well, where there's a lot of non-responses. And I've seen that in other trials as well, where there's a certain number that higher than we normally see in something like parasitoids or flies that don't respond. And we see that equally regardless of our treatment, but there is something I think to be explored there in tick physiological status, whether they're in questing mode or not questing mode, that we haven't really explored yet. That's a good question.
Thank you very much. Alvin, what about questions from there?
Yes, Marcelo?
Is Marcelo come to the microphone?
Yeah.
Yep. Hi.
Good.
Great talk. Thank you. Two questions. First, do you think you're planning to tell the Borrelia in attractivity of host first? And second, do you think that heat would be a key stimulus that needs to be present in order to return to host others?
Erika, did you get the entire question, or do we need to repeat because there was a little problem with the Wi-Fi there?
So I didn't get quite the first part of it. The
Okay, so
second part was
let's
about
have
heat,
let's
but-
have Marcel repeat that.
Okay,
please.
yeah.
Okay, my question is whether Borrelia might play a role in mice attractivity, because you mentioned that mice are very, very efficient in allowing infection to be acquired.
Yes. So I don't know, and that's an excellent question. I could get into that that aspect of I don't want to say sick animals, but challenged animals in some way. There have been some studies recently that have shown that Borrelia does not affect mice, and actually some that have shown that mice do better when they're infected in terms of survivorship and you know, a bunch of other ecological factors. So that doesn't mean that it wouldn't affect ticks, but we do know parasites do a lot of strange things to their hosts, and both in the tick itself and the host could definitely influence that system. The other part about the heat, we do monitor heat in that system, and we have had several iterations of the wide tube and or the 4-port where we have added or augmented heat, and it hasn't necessarily changed the outcome. If we add heat specifically to one area, we will potentially have some ticks attracted to that area with or without host odor. So the heat coming from the host in the air keeps that system pretty warm. So it's unlikely that is a factor, since both our treatment and our control sides are similar. We do augment both sides. Same thing with CO2. But it is a good point. And that's where we kind of get into that. Is it more close range? So do we have to be a little closer to these ticks for them to really perceive that heat? But it's an excellent point.
All right, thank you very much. Any question from the floor? There is
Nope.
Okay, go ahead.
There is no question from the floor
Okay.
yet.
All right. So also people from the speakers in the panel, if you have a question, you may want to unmute and ask the question. But I have a question here from Daniel. Did you analyze the chemical composition of the rodent waste? Maybe to know what chemical compounds were responsible for repellence?
Yes, so that's an excellent question. As I mentioned, as you saw, I didn't get into the chemistry a whole lot, but we have started to look at that a little bit, both host odors, just in general, what's coming off of these animals, and from the waste specifically. And I can give you just a little bit of a sneak peek that we have maybe 4 times the volatiles coming off of voles than we do mice. I mean, it's crazy what we have coming off of them. So it's not like they're that similar in terms of what they smell like or what's coming off of them. So I'm not sure what that means yet, but it would be very interesting to see, especially if we are legitimately getting some sort of behavioral response from these animals, whether it's movement towards questing or movement to go up or whatever, we may be able to manipulate that behavior in some way to some push-pull system or something along those lines. So it is interesting either way.
Carolina Reisman is asking, do mice and voles have a different grooming behavior that could explain the difference in abundance?
Yeah, so we don't know yet. That's what Jessica's looking at right now is their grooming behavior. What it seems like is happening from the animal side is mice groom more frequently, and they have a more standardized set of grooming behaviors than voles do. But voles have a stronger immune response. It's called anti-tick response to tick bites. So, like when somebody gets bitten by a mosquito and you itch, that seems they have a stimulus-driven response, and that tick bite tends to trigger them to groom more. So it seems like voles may have more of a role in this tick success because of that stimulus-driven response. We still have a ways to go with that, but that's where the data is taking us right now.
Any question from the floor there?
None.
No, no question there. Anyone from the panel want to ask any questions? We have run out of time, but we could afford one question. If anyone has one? No. All right. So thank you very much, Erika. Thank you very much to all speakers for the first part. We are here on time. And now we have a break of 20 minutes, a coffee break. I heard that the coffee breaks and the breaks in Kuala Lumpur are fantastic. We are so envious of you guys for attending person. That's the time for the break. Thank you very much. We resume at 4 minutes, whatever time is there. 7:40 is going to be here.
It's 10:40 here in Kuala Lumpur.
All right. Thank you.
Thank you, Walter.
All right, folks, welcome back. And thanks for attending the Symposium Frontiers in Chemical Ecology. The next speaker coming to us from the city of New York, the state of New York, but soon he'll be moving to New Haven, to Connecticut. Trevor Sorrells is the next speaker. Trevor, the floor is yours.
Hello, everyone, and thank you to Walter Leal and the other organizers of this joint meeting for the invitation to present in this exciting session. I will be talking about work from my postdoc with Leslie Vosshall at Rockefeller University. And this fall, I will be moving to Yale University to start my lab in the Department of Genetics. Mosquito behavior is responsible for the transmission of hundreds of millions of cases of human disease each year, leading to as many as 700,000 deaths. This constitutes a significant fraction of all infectious disease cases worldwide. Each of these cases begins when a mosquito senses, locates, and bites a human, and in the process transmits one of a number of different potential pathogens, such as parasites and viruses. The potential for the spread of these diseases is increasing with climate change because it is causing the range of mosquitoes and other vectors to expand. My research focuses on the question of what makes mosquitoes so effective at this behavior of locating and biting humans. We know a fair amount about how mosquitoes do this. They initially detect us using volatile cues such as carbon dioxide from our breath or odor produced by our skin. These cues cause mosquitoes to begin flying and tracking them upwind to locate their source. As mosquitoes get closer to us, they can detect additional cues, such as heat from our body and visual contrast, which they use to land on us and bite us. You'll notice that mosquitoes use a number of different sensory modalities to accomplish this behavior, and that it can occur over an extended period of time if they're not initially successful at finding us. So this raised to me the question of how mosquitoes combine these multiple different stimuli over time to accomplish this behavior. The work that I'm going to be describing was inspired by the fact that mosquitoes are micropredators, and we use this term because they consume only small quantities of their prey at a given time. But like larger predators, they use some of the same strategies to find us, such as tracking olfactory cues. Now, a number of studies in the mosquito and other flying insects have indicated how these animals will track odor plumes to their source. Upon sensing carbon dioxide, a mosquito will fly upwind, and upon losing the scent of carbon dioxide, they will fly perpendicular to the wind in order to once again locate the odor plume. And these responses have been documented on the order of a few seconds to a few tens of seconds. And this raised a question in my mind of whether mosquitoes had a hunting state that outlasts the response to these individual sensory cues. But to test this, I needed a way to deliver olfactory stimuli with high temporal precision. And to do that, I turned to optogenetics. With the help of a technician in the lab, Anjali Pandey, I created an optogenetic system for the mosquito Aedes aegypti that is responsible for the transmission of many viral pathogens. This system was designed to activate the carbon dioxide sensory neurons using light. And we used an existing reagent that captures the expression pattern of the carbon dioxide receptor, GR3. And we combined this with a red light shifted ion channel called CS crimson. And when these genetic elements are combined, it allows us to replace the delivery of carbon dioxide in airflow with delivery using light. And this provides us several advantages. First, when we deliver carbon dioxide in airflow, we have two stimuli for the mosquito combined, which are airflow, which is an important navigational cue, along with carbon dioxide. So this allows us to isolate the effect of carbon dioxide. And second, light gives us greater temporal and spatial control over the delivery of this stimulus. We tested these reagents in a number of ways, some of which I don't have time to tell you about, but the key experiment was to ask whether we could recapitulate the primary behavioral effect of carbon dioxide on mosquitoes, which is that when combined with heat, it causes mosquitoes to engorge on blood. And you can see that in this image here, where a heated blood meal in a glass chamber is being presented to mosquitoes, and the mosquitoes are piercing the membrane from below and engorging. And this experiment from past work shows that when you combine the heated blood with carbon dioxide, mosquitoes will engorge at high rates. However, if you present them just body temperature blood or carbon dioxide with room temperature blood, they will not engorge. To test whether we could recapitulate this with an optogenetic carbon dioxide stimulus, I created this assay called the Optomembrane Feeder that consists of a canister of mosquitoes receiving red light, and on the top of the chamber is a heated blood meal that is at 37 degrees. And you can see in this video that when we deliver the red light to these mosquitoes, they respond by flying around and eventually landing on this heated blood meal. And we quantified this attraction and found that these mosquitoes will respond to the stimulus when it is delivered either as red light or if we give these mosquitoes real carbon dioxide. And over time, the mosquitoes will engorge on this blood, which you can see in the video by their fattened abdomens. And we didn't see these responses in genetic controls where one of the two elements of this expression system is missing. And this demonstrates that mosquitoes are interpreting this red light stimulus in the same way that they interpret real carbon dioxide stimulus. So we refer to this stimulus as fictive carbon dioxide. Next, we turn to the question of how mosquitoes respond to brief human stimuli that more closely resemble what they would experience during actual host-seeking behavior. And to do this, I created an assay called the optothermocycler that consists of a plate of mosquitoes receiving red light from above for optogenetic stimulation and receiving heat from below that penetrates through the mesh of their container. And this heat is delivered using a PCR thermocycler so that we could rapidly change the temperature that they're experiencing. The output of this assay is video, which I processed by tracking 9 points on the body of the mosquito. And then I used supervised learning to classify which of a number of different behaviors the mosquitoes were exhibiting. And the software I used was developed in the lab of Kristen Branson at Genelia Farm. And when I start the video, you'll see that prior to the stimulus onset, the mosquitoes are primarily exhibiting grooming behavior. But then at time 0, they'll receive a 5-second pulse of fictive carbon dioxide. And you can see that they respond to this stimulus by flying, as well as walking and this behavior probing. And I'd like to draw your attention to probing, which is the term that we use for when the mosquito inserts its proboscis through the mesh at the bottom of the container. And this is a key behavior that they exhibit immediately before piercing skin. So we use this assay to quantify the response of mosquitoes to transient heat and fictive CO2 stimuli. Here I'm showing you an ethogram where each row is an individual mosquito, and the responses of the mosquitoes behaviorally are color-coded over time. And you can see in this graph of the mosquito's response to a 5-second heat stimulus, where the temperature goes from room temperature up to skin temperature in 5 seconds and then returns to room temperature. The response of the mosquitoes is to probe for a short period of time. When we tested the response of mosquitoes to the fictive CO2 stimulus, their responses were much longer and involved flying, walking, and probing. And in fact, the duration of these responses was vastly different, where the response to heat had a half-life of about 0.4 minutes, whereas the response to fictive CO2 only returned to baseline after about 15 minutes. And because of the importance of the integration of these different modalities of sensory cues, as I already told you, I presented these two stimuli simultaneously. And this resulted in a very strong probing response, but a reduced flight response. And this bias away from long distance movement of flight toward immediate attempts at piercing suggests that mosquitoes interpret these multiple host cues when combined as indicating the human is in their immediate vicinity as opposed to far away. And I was especially struck by the long-term response to fictive CO2 because I thought it resembled the hunting phase of predatory animals where they will stalk their prey over an extended period of time. And if this were true, one of the predictions you would make is that this behavior closely matches when mosquitoes have this goal of feeding on human blood. So to test that, we compared the response of females to those that had already taken a blood meal. After taking a blood meal, a female mosquito will spend several days digesting the blood meal. And during this time, she is no longer as attracted to human sensory stimuli. Similarly, male mosquitoes do not take blood meals, and that's because they don't need to produce any eggs. And so they don't, so similarly, they also do not show this strong attraction to human cues. So we compared normal hungry female mosquitoes to females that had been blood-fed. And we found that the blood-fed females had lost almost all of their attraction or almost all of their behavioral responses to both heat and the fictive CO2 stimulus. And when we compared male mosquitoes, we found that their response to heat was almost completely gone, but they had a strong response to the fictive CO2 stimulus. The difference between males and females was in the duration of the response, where females had a response that with a half-life of almost 10 times the length of the male response. And so this demonstrates that the sexual dimorphism in this behavior is not in the ability to detect the fictive CO2 stimulus, but it's in the duration of the response. And I was interested in learning more about the genetic basis for this sexual dimorphism. A graduate student in the lab, Nipun Boswar, had recently created a mutation in a known regulator of sexual dimorphism in flies called fruitless. And Nipun found that fruitless mutant male mosquitoes gained aspects of female behavior, including the attraction to human skin odor. And so I was interested in whether these mutant males would also have an altered response to the fictive CO2 stimulus. When I tested the response of fruitless heterozygous males, I found that it was very similar to the response of wild-type males. However, the fruitless mutant males had a stronger and more sustained response, particularly to fictive CO2. And this demonstrates that this regulator, fruitless, is responsible in part for this sexual dimorphism between males and females. Finally, I wanted to test whether this fictive CO2 stimulus could not only alter the behavioral output of mosquitoes for minutes afterward, but whether it could also alter their responses to subsequent sensory stimuli. And to do this, I created, with the help of a rotation student, Adriana Rosas, an assay called the blood blanket that consists of a thin layer of artificial blood located between the heating element of the thermocycler and the mosquito behind a mesh, so that the mosquito could access the blood, but we could also rapidly heat and cool this blood meal mimic. So we designed stimuli to deliver either a 5-second pulse of fictive CO2 or heating of the artificial blood for 10 minutes. And then we combined these stimuli and spaced them up to 20 minutes apart. Mosquitoes responding to these individual sensory stimuli engorged at low rates. And when they were combined, as expected, the mosquitoes showed higher rates of feeding. And we could space the fictive CO2 stimulus up to 14 minutes prior to the heating of the blood meal and still observe a potentiation of the mosquitoes' engorgement on the blood meal. And so this demonstrated that indeed the fictive CO2 stimulus results in not just an alteration of the behavioral output for minutes, but it also changes the response of the mosquitoes to subsequent heat and taste stimuli present in blood. And this is analogous to some sort of short-term memory, not in the same way of that we normally think of memory, but a memory that lasts for up to 14 minutes and may be representative that there's a human nearby. So to summarize, a brief sensation of human breath, in this case delivered as fictive carbon dioxide, causes mosquitoes to enter a state that lasts for somewhere from 10 to 15 minutes. During this time, mosquitoes show high arousal behaviors, such as flying and walking and probing. But this is not just a general arousal response. It is specific to the goal of feeding on humans. And we know that because if mosquitoes subsequently sense heat and taste cues, that they engorge at higher rates. And this persistent behavioral state provides a mechanism for mosquitoes to integrate these different modalities of sensory information over time. And we think it explains also why mosquitoes are so aggressive and effective at locating and biting humans. So finally, I'd like to make a small plug for my lab, which is opening this fall. We're interested in identifying the neural circuits that control blood feeding in the mosquito and understanding how they evolved in mosquitoes and some of their relatives. In particular, we'd like to identify the neurons that are responsible for this persistent behavioral state that I talked about today. We'd like to identify the neurons that both initiate as well as maintain the persistence of this state. And we think that these are going to be really important targets because they represent the difference between when a mosquito is actively seeking out a human and when they are not. And therefore, they may serve as potential targets to disrupt this behavior and to just understand more in depth how mosquitoes accomplish this. We're also interested in understanding how these neural circuits control mosquito behavior more broadly. In addition to the attractive stimuli that I talked about in my introduction, mosquitoes also deal with repellent stimuli such as swatting behavior of hosts. And we're very interested in how mosquitoes make these decisions about locating a human while still braving the danger of a defensive host. And finally, we will compare mosquitoes with some of their relatives that either do or do not feed on blood to understand how this behavior evolved and how it differs among these different clades of insects. And so with that, I would like to thank my postdoc advisor, Leslie Vosshall, as well as many members of the Vosshall Lab, some of which I mentioned throughout the talk. I'd also like to thank the rest of the Rockefeller community for providing a really helpful feedback on my project and helping with the design of these behavior assays that I used and many other technical and scientific advice over the years as well. And I'd also like to thank my funding sources. So thank you very much for this opportunity, and I'll be happy to take questions.
Thank you, Trevor. Very nice presentation. We already have a question coming in from Shefali. How prominent is walking response of mosquitoes in your behavioral assays, as I was under the impression that mosquitoes prefer flying? Also, if there was sufficient space for flying in your assay.
Yeah, thank you for that question. It's because of the nature of the assay, we used really confined spaces for the mosquitoes. And so they ended up, when they would fly for very short periods of time because they would hit the wall of the chamber and stop flying. But they did initiate flight events for about 15 minutes after the fictive CO2 stimulus. So even though they weren't able to fly long distances, they still had this drive to initiate flights. And I think that's less realistic for mosquito behavior, but it also has the advantage of decoupling ongoing flight from this internal motivational state for host seeking.
Any question from the podium there, Alvin?
Yes, there is.
Okay, let's go from there.
Dangshan?
Very interesting talk. Thank you. My question is, have you looked at the spectrum of the light? Which light has more effect than the other?
Yeah, that's a really good question, especially in light of recent work that is showing that mosquitoes show some attraction to red light. In my assay, I don't see a behavioral response to the bright flash of red light that I use for optogenetic activation. And the video is taken in the infrared spectrum, which there is also some suggestion that mosquitoes can see. However, I don't. I see a strong response behaviorally to bright green light, but not to this bright red light.
There is another question here as we prepare another question from the podium there. Would the potentiation by CO2 affect sugar feeding behavior? Consider there is CO2 modulation there.
Yeah, that's a good question. I didn't examine that specifically. I guess one thing that gets at is whether it's a general arousal response or is it specific to blood feeding. We don't see the same response to other arousal stimuli such as bright green light, which I mentioned, and I also did use an optogenetic sugar stimulus where you activate the sugar sensing neurons, and we didn't see as long of a response to that as we do for fictive CO2.
So there's another question from Berenbaum. Some or many flowers secreting nectar also release carbon dioxide. Could carbon dioxide response play a role in male food finding?
Yeah, that's an interesting question. Yeah, I have heard that suggested as a potential, evolutionary origin for host seeking as, from plant feeding insects that relied on CO2 from plants. In terms of male mosquitoes, I believe that they do use CO2 to track over long distances to get in the vicinity of hosts where they hope to mate with females. So they don't show the same strong attraction to land on skin that females do, but they do show this sort of long distance attraction to hosts.
Any question from the podium?
Yes.
Okay.
Dangshan?
It will be next, but my question is, it looks like you have a burst of light and that lasts 14 minutes. They have memory of 14 minutes. Have you tried to do that burst of light at different duration, see if that memory will last longer?
Yeah, I've done, I didn't do that comprehensively, but toward the, initial experiments, we did a lot of different durations, and, you do sort of see the longer stimuli show larger proportion of the animals responding, and, the same for intensity of light, but we didn't see a strong effect in terms of duration, but we didn't examine that systematically.
It seems there's another question from the podium there, but as they come to the microphone, let's take this one very quickly here. Just 1 minute. I'm curious, there is an advantage of using fictive CO2 over actual CO2 other than control for airflow. In addition to control for airflow, any other positive thing that you see?
Yeah, I mean, I think that is maybe the main advantage for studying sensory neurons, but one area that we'd like to go in the future is to target central circuits that can only be accessed optogenetically and can't be stimulated in any other way.
Okay, so Marcelo?
Yeah, thanks, Trevor. Very nice story. You referred to this as some form of a memory change. Could it instead be more of a motivational change? And then if this would be the case, could it be related to some change in biogenic amine levels after simulation?
Yeah, that's an interesting hypothesis. And yeah, memory is sort of, like a short-term memory or something. Maybe we lack a precise, a state change, on the order of minutes. I think that it could be some sort of neuromodulatory system that maintains that. And we don't know right now. There are similar persistent states in other organisms. So maybe, for instance, Drosophila, and we don't really know the mechanism that sustains them even in Drosophila. So maybe we'll get some clues over the next few years.
We are running out of time, Trevor, but can we address this question from Greg? Wood Foster has done a lot of work looking at the behavioral switch from sugar feeding to host seeking in females. Have you looked at the lack of fictive CO2 response in females post-eclosion?
Oh, that's an interesting question. I have not looked at that. One confounding variable we might have there is the expression level of the optogenetic channel, because that may depend a little bit on age of the animals. So immediately post-eclosion, they may not have as high an expression level of the channel. So it might be hard to do that way.
Thank you very much. We might come back to that later. Now we have to move the podium to Blacksburg in Virginia Tech, where Chloe is going to do her presentation to bring the latest from her lab and share with us here. So let's go to Chloe. The floor is yours now, Chloe. Go ahead.
Hi, everyone. My name is Chloé Lahondere. I am an assistant professor in the Department of Biochemistry at Virginia Tech. And today I'm going to talk to you about a project that deals with the effects of personal care product use on mosquito-host interactions. This project was led by Morgan van der Gissen, Anaïs Talon, Brenda Miko, and is a collaborative project with Clément Vinoget, also at Virginia Tech. So why do we study mosquitoes? Mosquitoes can transmit many pathogens to humans, causing diseases such as Zika, dengue, malaria, and yellow fever. And unfortunately, these diseases can cause the death of more than 1 million people per year. There are many challenges that are associated with mosquitoes as well. Vaccines and treatments are not always available for a lot of these different diseases. There is a rise in insecticide resistance that is challenging current control strategies. Some species are outsmarting our control tools. There is, for example, this mosquito species that is nocturnal but starts biting during the early morning to avoid bed nets. And then finally, there is a big problem with invasive species and climate change. So we need new solutions and tools to limit mosquito populations and prevent pathogen transmission. So mosquitoes, like many other insects, use olfaction, among other cues, to locate hosts to feed on. Olfaction plays a critical role throughout their life. After emerging from the pupae, males and females are going to be using plant odors to obtain a nectar meal. They are then going to be using conspecific odors to find a mate. Females are going to be using odors emanating from the host to obtain a blood meal. And once they are ready to lay their eggs, they look for oviposition site odorants. Interestingly, we also use plant odors in our daily life. If we look, for example, at the use of soap, of shampoo, of detergent, of perfume. Just as an example here, a brief history of soap. The earliest recipe that is known is from the Sumerian civilization almost 5,000 years ago. People from the ancient Egypt civilization used a lot of these different plant-derived compounds for creating balms, ornaments. They also use waxes in their rituals. The more like modern soap emerged during the medieval Europe, and thinking, for example, about the Savon Marseille, this recipe is almost unchanged and is still used right now. Hygiene has drastically increased during the Renaissance and people starting wearing more and more perfume. And then we need to wait for the Industrial Revolution during the 19th century to see really a boom in the use of soap. At this time, it's mainly hard soap or bar soap. But then a little bit later, during the 20th and 21st century, we see the invention and emergence of liquid soap that is going to change a lot, because we can use it for like shampoo, detergent use, hand soap, and body washes. And today we have access to such a large range of different products, some of them being more naturally derived and some of them being more chemically derived. And if we look at just a couple of examples here of the composition or ingredient lists of some of these body washes, what is fascinating is that they are full of mosquito semiochemicals. So plant-derived odorants, for example. I'm thinking about linalool, citronellol, geraniol, limonene. A lot of these different products also contains extract of plants, essential oils, and a more obscure term, which is fragrance. It's basically a blend of different compounds. In that case, that's under the name of perfume. So we can see that these body washes that we use every single day are full of mosquito semiochemical. So then this question arises: what is the potential impact of this soap use on our scent, on our body odor? We know that mosquitoes use our different compounds that are emanating from our skin to locate us and to bite us. So how is it affecting their mosquito host-seeking behavior? And what is also interesting is that a lot of different research conducted on chemical ecology and mosquito-host interaction is led on volunteers that are unwashed, or we ask them to wash with a specific soap. And this is also the case in our lab. But we know that every morning we are using a lot of different like creams and hand soap and shampoos and body washes. So the hypothesis for this work was that soap application would alter the host profile, the host odorant profile, maybe adding some VOCs to the headspace, or maybe removing, masking some of the naturally occurring compound on the skin. We had several objectives for this project. How does soap use affect the host chemical signature? Do all soaps affect the host chemical signature the same way? What are the potential effects on mosquito host-seeking behavior? And can we use that information for the development of new baits and repellents? So our first aim was to test whether soap use would affect our chemical body signature. So for that, we selected 4 different soaps based on their brand popularity and composition, making sure that they contain some of the semiochemicals I mentioned earlier. We took 2 more chemically derived soaps and 2 more natural derived soaps. And what we ask our volunteers to do is to wear 4 different sleeves on their arms. So on one side, they were wearing 2 sleeves on the arm that was unwashed, and then the other arm was washed with one of the soap that we provided them. And then using SPIMI, we collected the volatile organic compounds emanating from those sleeves, and some of these samples went to our GCMS to analyze the composition of these VOCs, and the other sleeves went to our behavioral cage assays, where we tested the landing behavior of Aedes aegypti female mosquitoes. So here are representative chromatograms for our different volunteers and the different soaps that we use. We're going to take the example of volunteer number 1 here and compare the chromatograms before they washed and after they washed. And what we can see is that first, after washing these two first kinds of soap, you can clearly see that there is an increase in the number of chemicals that are present in the chemical profile, and there is also an increase in the abundance of those chemicals. It is actually interesting because it's dependent also on the host here. You can see that the effect of this second soap leads to, a smaller variation in their chemical profile compared to, for example, volunteer number 2. In the case of the more like natural derived soaps, you can see that the effects is different, and some of them also lead to just like an increase of abundance of these chemical compounds and existing, or just like adding some of them to the scent of the host. So what we found is that soap wash actually effectively affects the volunteer chemical profiles, adding some chemicals or removing some of them and increasing the abundance of existing chemicals present in the scent. There are some variations that were observed between soaps, but also between volunteers, obviously. Now, when we plot these different samples based on chemical abundance and composition, you can see here a big cluster of all of volunteers' samples that were unwashed. And then you can see that it's clustering pretty well away from these two different kind of soaps that are the more like naturally derived soaps versus the ones that are more chemically derived soaps. So there is a statistical impact, statistically significant impact of the soap use here on the host chemical profile, but this is dependent on the soap that is used. Now, if we look at the relative abundance of these different functional groups here, what we can see is that for the unwashed volunteers, their body scent is dominated by aldehydes and ketones. But once we ask them to wash with these different soaps, you can see that this is clearly shifted towards a more prevalence of the terpenes in the scent. Again, this is the relative abundance. So the host chemical composition is clearly shifted, but there are like variations also between the different soaps. Here you can see that it's dominated by pseudolimonene and linalool, but this is changing depending on the soap that the volunteers used. Now, if we look at the absolute abundance, so the concentration of these different compounds in the scent, you can see here, and we're going to zoom later on the different categories, but for the unwashed, you can see that the relative abundance is pretty low for all of these different compounds or family of compounds. But once we ask to wash with this soap, this first soap or the second one here, there's clearly like shifted an clear increase in aldehydes, ketones, but in particular terpenes. And in the case of these two more naturally derived soaps, you can see that is clearly a shift towards the terpene. So now we know that there is a clear effect of soap use on changing the oral chemical signatures, but how does it translate and affect the mosquito host-seeking behavior? So to test for that, we tested the landing behavior of mosquitoes in cages where we put two different sleeves. In the first case here, we tested whether mosquitoes would have a preference for a sleeve that was not worn by a volunteer. So in that case, you can see that they are not attracted to either side of the cage and that the proportion of landing is fairly low. After that, we tested whether our volunteers were attractive to the mosquitoes or potentially aversive to the mosquitoes. And what we found is that the mosquitoes were landing more on the sleeve that was worn by the volunteers. And after that, we tested the effect of soap use. So in that case, the mosquitoes had the choice between the sleeve that was worn by the volunteer that had washed their arms or that did not wash their arms. And what we can see is that the mosquitoes tended to land more on the side where the soap was used. It was true for the two more chemically derived soap, as well as significantly higher in the case of this first type of natural soap here, the proportion left landing being obviously quite high. Interestingly, for this last naturally derived soap, we can see that it led to an aversion. So mosquitoes tended to land more on the sleeve that was on the arm that was not previously washed. So there is also a clear effect in the valence of the soap use changes depending on the type of soap. Our next question was, can we inform, and we use this data to inform the development of new baits and repellents? So we took an analysis that is usually used in the field of ecology, where people are testing the association between different species with different habitats. In our case, we have different types of chemicals that are associated with the behavioral output. And the aim here was to identify chemicals that were significantly associated with a host-soap combination, and that resulted either in a strong and significant attraction or aversion. And so we recreated artificial mix based on that. We created an attractive mix and an aversive mix. And we tested these again in our landing behavioral assays, like I presented before. But in that case, mosquitoes had access or choice between a sleeve that was born on an arm that was not with the attractive or aversive mix, or an arm on which the sleeves had been, we added the attractive mix to. And what you can see in the case of the attractive mix, the mosquito preference was really clear, and the proportion of landing was quite strong. When they had access to just two different sleeves worn by the volunteer, they didn't show any preference and landed on both the same way. And in the case of the aversive mix, they were landing more on the side that did not add the aversive mix to it, meaning that we can indeed use that knowledge to inform the development of attractive mixes and aversive mixes. So to summarize our findings, we saw that soap use alters the host chemical signatures. There is a modification of the odorant profiles for all of the different volunteers that we tested. There is a clear shift in the relative abundance, particularly of aldehydes and ketones, with the addition of terpenes once they have been washed. The soap use affect the mosquito host-seeking behavior clearly. There is a host and a soap dependence, and it can lead to either an increase in the attraction or the aversion for the soap side. This knowledge can definitely be used to create new baits and repellents, as well as potentially develop new personal care products that might have some repellent properties to protect hosts from mosquito bites. So this project is a kind of a proof of concept project, but what we are working on right now is to try to understand how long that shift in the odorant profile lasts after the application of soap. We are also wanting to know how factors such as heat and humidity could affect the host chemical profile after the use of PCP or personal care products. And finally, we know that our scent is, partially due to the microbiome metabolism. And so what we want to know now is that how our microbiome interact with the use of these personal care products. So I would like to thank Dr. Walter Leal for inviting me to participate in this amazing symposium. I would like to thank also my co-authors, Bryn, Anaïs, Morgan, and Clément, as well as our insectarium technicians, Sajida Diggs and Sayed Miroli. I would also like to thank our funding sources, and I would like to acknowledge that I work on land that belongs to the Tutelo Monacan people and that I respect their nations. And I would like to thank you all, and I would be happy to take any questions. Thank you.
Thank you very much, Chloe. So let's open the floor for questions. There is one already here. Let me see before we have the question from the floor there. Interesting work. We showed native show repellent. What do you think are the chemical contributors for the same?
That's a good question. So that's what we did in the last part, where we actually, with the statistical analysis, we extracted 4 different compounds that were responsible for the aversive behavior of the mosquitoes, and we took those to put in our blends. But this specific soap contains coconut oil, and we think that it has been shown to be partly repellent to some mosquito species, and we think that is also driving some of this aversion.
What about the floor there? Any questions?
Yes, there is. Manish?
Yeah. Hi, I'm Manish from India. I have a basic question. Like, you have used head space of human, like washed with soap. Is there, have you checked at the effect of only the head space of soap? How like that will attract and repel the mosquitoes?
That,
Like how
that's
does soap's headspace behave?
That's a very good question. This is not something that we tested just because the concentration was like kind of the concentration that we have on our skin after application is radically different from what you get directly from the bottle. So we think that probably mosquitoes would be like repelled. That's at least, you know, some kind of hypothesis that we have, but we did not test it directly, the soap in our assays.
The ISCE congratulates you for the work and they ask, can you elaborate or explain what was control unwashed and how long were the volunteers unwashed, or were they all washed and wait for certain pre-exposure time? A little bit elaboration on the method.
In the methods. That's a good question too. So what we asked the volunteers to do was to wear the same day, obviously, these false sleeves, and on one side, on both sides, like we asked them first to do an ethanol wash. So basically removing any potential soap traces from their own, you know, soap use at home. And then we had them wear those sleeves for half an hour. And they had to wash with a specific volume of soap, a specific dilution of soap, each of them washing for the same time, the same way. So we asked to have them just like regularly and just like be exactly the same across different participants. And what we did was also to replicate that. So we did several behavioral assays for these different hosts and at different time, right? So we did that over the span of several months just to see if we could have any potential impact of like variations in the same composition of the host, even just the unwashed host, basically hosting nature.
Any question from the floor, Alvin?
None.
None. As we wait for more questions, if they're coming, I have a close interest in this work because my daughter, when she was small, she always complained that the bees were always coming to her. And I never believed that very strongly until one day when she came to the lab and did some work with us, and she had to wash. And one day for the skin to get the composition, look at the mosquito behavior. One day she had an extract, and I got it. I still have the data here. I got a very big peak of isoamyl acetate, which is a compound that attracts the bees, in fact, but it's cause a stink behavior of the bees. So she was wearing this shampoo, OC, and I told her that's where it comes from. Now I believe it, and she stopped using that shampoo and never had problems with the bees anymore. It's interesting because you are right on target. Whatever you use might have an attractive or repellent effect. So Daniel asks here, he says, great research, most soap contain perfumes. How did you handle the influence?
Yeah, that's a very good question. That is so obscure. So even if you get a more like naturally derived soap with like less ingredients in the list, but each time you get fragrance or perfume, this becomes a nightmare just because looking at those chromatograms, you can see that there are like a lot of different peaks and a lot of these different compounds that we find in the other soaps as well, like limonene, linalool, all of those like compounds, even if they are like naturally derived, these are still compounds that are semiochemicals for mosquitoes. So this is something that is really hard to handle. And I think it's also part of the, like each of these brands have like proprietary, you know, recipes that they are trying to, you know, keep. And so they don't want to give off, you know, a lot of details about the composition. But for us, it's like, it makes it a little bit more difficult. But the GCMS is not, you know, it's telling us what we want to know, which is pretty convenient.
We're almost there on time. Are there still questions from the floor there, Alvin?
There is none, Walter.
No question from there. And apparently no question from here unless I missed something. It's very interesting work. Keep the good work going, and we might have some question later. So let's move on now to the next speaker. Now we go to Baltimore, Maryland, to Johns Hopkins, where Ali Afif is going to tell everything about the DT response by mosquitoes. and he's Johns Hopkins, but soon he's going to be moving to Philadelphia to start a new job as assistant professor at Drexel University. Afif, the floor is yours.
Hello, I'm Ali Afify, and today I'm going to talk about the work I did during my postdoc at the Bartle Lab on mosquito repellents. Surprisingly, their mode of action is not well understood. Some repellents work by contact, meaning that mosquitoes land on a skin treated with the repellent, touch the repellent, and then get repelled. But this does not explain why in most cases mosquitoes do not land on the skin treated with the repellent. This suggests an olfactory component. This simple behavior, mosquito repellent avoidance, can be explained as all behaviors by neuronal activation. In this case, the hypothesis is that olfactory neurons get activated, and this leads to mosquito avoidance to repellents. Neuronal activation can in turn be explained by genes, or the expression of odorant receptors in olfactory neurons. And this is what I'm interested in and what I'm going to talk about today. I will first talk about the mode of action of mosquito repellents at the neuronal level. Then I will revisit mosquito behaviors towards repellents in light of my recent findings. And finally, I will talk about odorant receptors that are responsible for this behavior. But before I do this, I will first introduce the mosquito olfactory system. This is the mosquito nose, the antenna. It's covered with many sensory hairs called cincella, and each of these cincella contain odorant receptors, or ORs. ORs are always present with their co-receptor ORCO. ORCO is required for OR function. Previous research in Aedes mosquitoes found that deleting ORCO leads to mosquitoes not able to detect the insect repellent DEET. So this suggests that DEET is sensed by Aedes mosquitoes by using ORs. In Culex, an OR was found to be activated by DEET, OR136. How about Anopheles mosquitoes then? Is it also using ORs to detect DEET and currently use mosquito repellents? To answer this kind of questions, researchers have been using a technique called single cincellum recording by inserting an electrode into a cincella and measure its electrical activity towards repellents or odorants in general. But this is a challenging technique, especially in mosquitoes, because it has a fragile antenna, and also because the antenna is covered with many cincella that we need to test each one of them. Therefore, we express the calcium indicator GCAMP into the olfactory neurons of Anopheles mosquitoes. Now, once an odorant activates an olfactory neuron, GCAMP in that olfactory neuron shows an increased fluorescence. Now we can image the mosquito antenna while delivering a 1-second pulse of an odorant into an airstream directed at the antenna and measure the responses of the antenna neurons towards that odorant. And this is how odorant-induced responses look like in the mosquito antenna. For analysis, we decided to focus only on one antennal segment because it's representative for the responses of all the antennal segments. Now we can use this method to test many insect repellents, and this is a list that we already tested. It contained man-made repellents like DEET, IR3535, and Bicardin, and also natural repellents like lemongrass oil, eugenol, and p-methane diol. And here, this is an example of imaging recording in mosquito antenna using lemongrass oil at 1%. As you can see, once lemongrass arrived to the mosquito antenna, it elicited a strong response in 3 to 4 neurons. We can turn this into heat maps to show the position of these responding neurons on the mosquito antenna. Then we tested PMD, and also it elicited a strong response in about 4 neurons, and eugenol, which elicited a very weak response. How about synthetic repellents then? We used this method to test DEET and IR3535 and Bicardin at different concentrations, and we found, for our surprise, that DEET and IR3535 did not elicit any response even at 100% concentration, while Bicardin elicited a very weak response and only at 100% and not in lower concentration. So how these synthetic repellents work then? In the natural situation, we spray these repellents on our skin, so mosquitoes encounter them as mixtures with our own odorants. So we wanted to mimic this natural situation by testing synthetic repellents mixed with our own odorants. And we started with 1-octane-3-ol, a component of human skin odorants that is also attractant to mosquitoes. This is the response to 1-octane-3-ol, also a strong response on the mosquito antenna neurons, but when it's mixed with DEET, IR3535 or Picaridin, this response was almost completely gone. We think that this means that these synthetic repellents mask the response to 1-octane-3-ol. How about other human skin odorants? Will they also behave the same way, or this masking is only related to 1-octane-3-ol? So we tested another 5 skin odorants, including 2-acetylthiophene and benzaldehyde, and as you can see, all of these skin odorants were masked by DEET at different concentrations. And we found that at least 30% of DEET is required to completely mask the response to these odorants. This could explain why these synthetic repellents, DEET, IR3535, and Picaridin, are used at high concentrations, around 30% in commercial repellents. Now, how this masking happens, how DEET and other synthetic repellents inhibit odorant-induced responses. There are 2 hypotheses here. One, that DEET work on the receptor level by binding to the receptor, preventing human odorants from reaching the receptors and binding to those receptors. The other, that DEET works on the chemical level by binding to the human odorants itself and prevent it from activating odorant receptors. To test these hypotheses, we decided to modify our odorant delivery method. Instead of delivering 1-second pulse of an odorant towards the mosquito antenna, we deliver 3-second pulse of DEET or the other synthetic repellents, and then followed by 1 second of the human odorants 1-octane-3-ol. In this case, if DEET work on the receptor level, it will arrive earlier and it will inhibit that receptor, preventing 1-octane-3-ol from binding to the receptor. But this was not the case. We found that octanol, whether it comes after paraffin oil control or after DEET, elicit the same response. And the only decreased response is when we mixed 1-octane-3-ol with DEET as physical mixing. This suggests that the DEET and the other synthetic repellents work on the receptor, on the chemical level, not on the receptor level. And this is probably by decreasing the amount of odorants that can reach the mosquito antenna. To confirm this, we used a photoionization detector, or PID, with calcium imaging to measure the amount of odorants that reach the mosquito antenna while doing imaging. So on top, I will show imaging data, and on the bottom, I will show the concentration of the odorants that reach the mosquito antenna while doing these imaging experiments. So here we tested DEET at first, and as expected, DEET did not elicit any response on the mosquito antenna, and also it was not really detected at the mosquito antenna by the PID. When I tested 1-octane-3-ol, it elicited a strong response in mosquito antenna and also was detected at higher concentration around the mosquito antenna. And then I tested the mixture of 1-octane-3-ol and DEET, and the responses on the mosquito antenna was almost completely gone, and also the concentration of 1-octane-3-ol decreased significantly that reached the mosquito antenna, which suggests that DEET indeed prevents 1-octane-3-ol from reaching the mosquito antenna. So this is a summary of this part. This is our model of how insect repellents work with Anopheles mosquitoes. First, we have repellent activators like lemongrass oil and PMD, which evaporate, reach the mosquito antenna, drive these mosquitoes away by activating odor receptors on the mosquito antenna. We have also repellent maskers, which are synthetic repellents that do not really evaporate as much, but they also prevent our own odorants from evaporating and reach the mosquito antenna, and this leads to a lack of attraction, so not really active repulsion as we think it is. So now we talked about neuronal activation, how this translates into behavior. To test mosquito behavior through repellents, available methods like the Armin-Cage assay or the Y-tube olfactometer were used. In both methods, repellents are mixed with human odorants to see if these odorants still attract mosquitoes. But here, according to my calcium imaging results, some repellents do not activate antennal neurons and might not have a direct effect on mosquitoes. And I wanted to test this. Do these repellents really lose their effect in the absence of human odorants? So we designed a new assay. And we called it the Close Proximity Response Assay. Simply, we introduce a single mosquito into a cage and we leave it until it lands on the net wall of the cage. Then we approach that mosquito with a pipette tip that contains an odorant or a repellent and monitor that mosquito for 30 seconds to see if the odorant repels that mosquitoes away. And then we tested 3 mosquito species using that assay. Anopheles, which according to our own result should not be repelled by DEET. Aedes and Culex, which according to previous research should be repelled by DEET because it activates odor receptors on the mosquito antenna. So we did that. This is the responses to Anopheles mosquitoes. What you see is a survival plot showing the proportion of mosquitoes that remains on the wall of the cage, which are the mosquitoes that did not respond to the repellent. The black line shows the response to paraffin oil, and this is the response to DEET, IR3535, and Picardy. As you can see, they did not elicit any response in the Anopheles mosquitoes. Anopheles mosquitoes did not fly away in response to those 3 synthetic repellents. Then I tested natural repellents, and as you can see, this is the response to, in red, the response to lemongrass oil. In blue, this is the response to PMD, and then the response to eugenol. As you can see, lemongrass oil and PMD elicited a strong response, drive mosquitoes away, but eugenol did not, which also agrees with our calcium imaging results that eugenol elicited a very weak response on the mosquito antenna. Now, Aedes also natural repellents, including eugenol, elicited a strong response, drove mosquitoes away, and also DEET elicited a repellent response in Aedes mosquitoes. The same for Culex mosquitoes. DEET and the 3 natural repellents elicited a response in Culex mosquitoes. This confirms our own hypothesis that DEET works only in Culex and Aedes mosquitoes, but does not work directly in Anopheles mosquitoes. So this is a summary of our behavioral experiments. We showed that DEET directly repels Aedes and Culex mosquitoes, probably by activating odor receptors. I also showed that in Anopheles, DEET works only by chemically masking human odorants and not directly repelling Anopheles mosquitoes, which leads to a lack of attraction. I also showed that natural repellents directly repel all 3 mosquito species. So now, which odor receptors are involved in the direct repulsion by natural repellents? Calcium imaging shows which neurons respond to an odorant, for example, lemongrass oil, but it does not tell us which ORs are expressed on those neurons responsible for their activation by lemongrass oil. To answer this question, I decided to cut around those responding neurons and run RNA-seq to identify odorant receptors responsible for their activation. But this is challenging, first because the responding neurons change their position between mosquitoes, and also because the tissue is big enough that it contains non-responding neurons in addition to the responding neurons. But we reasoned that if we run this multiple times, we will enrich for the responding neurons, so we can get a hit list of neurons that might be responding to lemongrass oil. So I did that in 12 samples of lemongrass oil. And the same for PMD. I also tested 12 samples of PMD followed by RNA-seq. And this is a summary of our RNA-seq data. Unfortunately, 1 sample from lemongrass oil and 2 samples from PMD did not show any RNA. As expected, Orco was found in all samples. OR15 and OR9 were also found in all samples, while all the other ORs in the list were found in most samples. So this is our hit list that contains the ORs that are present in at least most of the samples. Does this mean that these ORs respond to lemongrass oil and PMD? No, but it means that they are most probably contain the ORs that respond to lemongrass oil and or PMD. So we decided to use the empty neuron system to test which of those ORs actually respond to lemongrass oil and PMD. Basically, an OR, OR22A, is deleted from the AB3 syncylla on the Drosophila antenna. Now, a neuron is empty, does not contain any OR, and we can express any of the Anopheles ORs into that neuron, and then test that using the single syncylla recording by inserting an electrode into the syncylla and measure its electrical activity towards odorants. And this is an example of the responses in the AB3 syncylla, which contains two neurons, the A neuron, which is the higher amplitude neuron that express the OR from Anopheles, and then the lower amplitude neuron that contains the Drosophila OR, which is not really interesting for us. And this is an example of an OR, OR20, that responds strongly to lemongrass oil and also to PMD. And this is another example of an OR, OR10, that does not respond to lemongrass oil or to PMD. And this is a summary of the responses of all ORs we tested so far. As you can see, OR20 responded strongly to lemongrass oil and PMD, while OR9 responded strongly to PMD but showed a medium response to lemongrass oil. Here you can see also in red that some ORs we didn't test yet. These we had to actually make the lines, and now we are ready to test them. Now, in order to find out how much these ORs are involved in shaping mosquito behavior towards the two repellents, we will make mutants of those ORs and test them in the close proximity response assay to show whether they are actually required or not for the repellency towards lemongrass oil and PMD. To summarize, I showed that lemongrass oil and other natural repellents activate olfactory neurons on the Anopheles mosquito antenna, and this leads to these mosquitoes flying away from lemongrass oil in the close proximity response assay. On the other hand, synthetic repellents like DEET do not activate olfactory neurons on the mosquito antenna and also do not repel Anopheles mosquitoes away in the close proximity response assay. These synthetic repellents work only by masking the response to human odorants like 1-octane-3-ol. I also showed that odorant receptors like OR20 and probably OR9 are the ones responsible for the response towards lemongrass oil and PMD. In the future, I would like to use the same methods to test other mosquito behaviors like oviposition behavior to see how mosquitoes respond to oviposition cues, and also to test whether oviposition cues activate olfactory neurons on the Anopheles mosquito antenna, and also to find which odorant receptors are involved in this process. This work I will do in my lab, which I will start at Drexel University next month in September. Finally, I would like to thank all current and former members of the Potter Lab, as well as our collaborators, Chloe Lahondere and Joshua Pitts. I would like to also thank the Department of Neuroscience at Hopkins, the Johns Hopkins Malaria Research Institute, the NIH, and the DoD for funding this project, and you for listening. Thank you. Thank you.
Thank you, Ali Afif. A very nice presentation. The questions are already coming. One here from Shefali. Nice work. How separate was the delivery of octane-3-ol and the DEET?
Thank you. This is a good question. Actually, they were not separated at all. So DEET starts, and then after 2 seconds, 1-octane-3-ol, while DEET continued for 1 more second. So there was an overlap between DEET and 1-octane-3-ol. And we did that intentionally to make sure that that DEET is still going to the antenna while delivering 1-octane-3-ol.
Any question from the floor in Kuala Lumpur?
Yes, Renee, to the mic, please. Thank you.
Can you hear me?
Yeah.
Yes, go ahead.
Right. So I was curious in the ecological reasons for why lemongrass is a repellent, either ecological or evolutionary. I mean, lemongrass contains compounds that are detrimental to mosquitoes, are going to actually, kill mosquitoes. So what is the basis for the repellents? Any ideas?
We are not sure. We didn't test this. I think it might be that lemongrass oil might work as lethal to mosquitoes. So, for example, we found that also mosquitoes would avoid laying eggs on lemongrass oil. So apparently, it might be lethal for the larvae, and that's why they don't want to lay their eggs on lemongrass oil. I don't know, it might be something evolutionary that because it kills mosquitoes, they avoid it. But lemongrass oil so far is probably the strongest mosquito repellent, at least for Anopheles, that I tested.
There are two questions. Oh, okay, go ahead. In follow-up on that, even? No? All right, so there are two questions that are somewhat related. So do many chemicals besides 1-octane-triol get affected when mixed or delivered with DEET? And the other question, I think they're part of the same type of question. Is the masking behavior of DEET observed for any other ants other than octane-triol? How was it vary with concentrations?
So for the first question, yes, we tested 6 human odorants, including 2 acetyltyphene. So these are chemicals on our skin that mostly attract mosquitoes. And we tested all these chemicals, and all of them were masked by DEET and also the other 2 synthetic repellents, IR3535 and Picaridin. So this masking effect happens in at least those 6 chemicals, 6 human odorants, not only 1-octane-triol. I'm sorry, can you repeat the second question?
The second question is the mask behavior of DEET observed for any other odorants other than octane-triol. How was it varying with concentration? I think there are 2 separate parts of the question.
Yeah, so we did not test different concentrations of the human odorants. We tested a 1% concentration of all human odorants, but we tested different concentrations of the repellents. So from 1% to 30%. And we found that masking happens even at the lowest concentration of the repellents, but at 30%, at least 30% concentration of the repellent is required to completely mask and cancel the effect of the human odorants. And this would explain why these synthetic repellents are used at very high concentration, around 30% in commercial repellents, because you need that to actually prevent a mosquito from smelling human odorants.
If there are no questions from the floor, I will have one from the chat in here. No question from there, right? So interesting, this says Jin Zhang. I'm curious about how much tissue you cut after your single-cell record experiments to do the RNA-seq. And a part of the antenna segment or just a surface tissue? Was that a part of the segment or just the surface tissue?
So yeah, we actually tried different methods. So I wanted first to actually cut just around the neurons that are activated. And we used laser capture microdissection. But because this method is actually used with tissue cultures mainly, and when we did that with mosquitoes, it actually cut and destroyed the whole antenna. So I decided to do that manually, and I was able to cut 1/3 of each segment. So 1/3 of the segment, so not only the surface, but just 1/3 of the whole mosquito segment. Yeah, so that was the smallest tissue that I could actually manually dissect.
We're almost run out of time, but there's one more question. Here's come from Zain. How comparable are response across the segments? And were all the Sensilla basiconica type or trichoid?
So for the, I could say that between segments, I could see the same responses or similar responses. I'm not saying like the number of neurons, but I'm talking about if there is a response or not, and if there is masking or not. Because we did whole antennal recordings, and we saw that the responses are similar between all segments. The Sensella, we did not test different Sensella, and we don't really know because we were doing imaging. So we were actually testing or recording from the neurons, not from the Sensella. So we could not see actually which Sensella are actually activated, but we could see the neurons on the main antenna, so not through the Sensella. So what we see is actually the cell bodies of each of those neurons that get activated.
Very quickly, this is where I almost run out of time. There are the good questions that keep coming, but let's just answer one of them, okay? Yeah. Where all the lemongrass oil responsible olfactory receptor neurons typically clustered together, or did some of your tissues need to say that, let's see here, discovering my thing, consists of large segments?
Yeah, so
I need to repeat that.
Yeah, most of the samples that I got was actually clustered together. So the neurons were actually clustered either to either sides of this mosquito antennal segment, except for like one sample that I saw mainly in all of the antennal segment. The neurons were just diverse on all the antennal segment. But most of them were just clustered around one side of the antennal segment.
I got a waiver here for another question, but very quickly, does DT act by reducing the vapor pressure of the odorant? Would a polar compound that doesn't interact with DT avoid masking? That question comes from Trevor, by the way.
Yeah, it's a great question, actually. I think this is one of the, solving the mystery of how DEET actually does this kind of masking would be really important, but we really didn't go into that. We just found that it prevents those odorants from evaporating, but the mechanism of how DEET actually does this, we don't really know. We tried using GCMS to do this, and unfortunately, DEET is a very heavy compound that we could notice in samples that does not contain DEET after about 1 week of using DEET. That's why it's a very tricky compound to actually test. So yeah, I don't know how it works. It's just decreased the amount of evaporation. So maybe it just decreased the vapor pressure as Trevor suggested.
Thank you very much. We will go back. There are more questions, but we need to move on in the interest of time. And I mentioned in the plenary lecture that I gave you before this symposium, a ground-based elucidation of the first structure of an odorant receptor. It happened to be the odorant receptor core receptor, also named as ORCO. And the person who authored that paper, the first author of the paper, is Joel Butterwick, and he's joining us today to talk about odorant receptors, not necessarily what he did in that paper, because we already read that paper, but what he's doing now in his laboratory at Yale Department of Pharmacology as an assistant professor. Please join me and welcome Joel Butterwick. Joel, the floor is yours. Go ahead. Glad to have you here.
Hello. I'd first like to thank Walter Leal for inviting me to participate in this symposium today. My name is Joel Butterwick, and I'm a new faculty member in the Department of Pharmacology at Yale University. So we've heard a few stories today about how critical olfactory signals are to insect behavior and survival. And now in my laboratory, we're interested in understanding sort of the biophysical principles that underlie chemosensory receptor function. And so for my talk today, I'm going to focus on how olfactory receptors function really at the molecular level and discuss two unpublished stories. The first is going to focus on understanding how the insect olfactory receptor co-receptor ORCO has evolved to remain inert and not contribute to olfactory receptor tuning. And the second story is going to be about how some molecules can overcome this evolutionary pressure to activate ORCO and thus most of the olfactory system. So the chemical world around us is vast, and it is incredibly diverse. And as you can appreciate here, odorants and tastants have discrete molecular structures that differ with respect to a variety of physiochemical properties, each one contributing in a yet unappreciated way to their different smells and tastes. Now, olfactory and gustatory systems are thus faced with the same fundamental challenge to accurately describe an almost limitless and discontinuous chemical landscape, where even a small change can have drastic implications for an animal. And so to contend with such complexity, chemosensory systems across animals essentially use a common strategy that relies on large families of receptors, often numbering in the hundreds or thousands in a given species. And this is in contrast to other senses which rely on only a few receptors. For example, 3 receptors are sufficient to detect the entire spectrum of visible light, allowing us to see. So in the olfactory system, each receptor is sensitive to a different series of odorants, which will often overlap with other receptors. Consequently, when we smell, odorants will activate a subset of receptors, and this sort of combinatorial activation of receptors is used by the brain to distinguish which odorants are detected. And this is really an elegant mechanism, but it requires that the chemical sensitivity of each receptor is different from the others. So now insects have evolved a unique molecular solution to detect odorants. They utilize a novel family of chemoreceptors with no homology to any other class of molecule. And these chemoreceptors are ligand-gated ion channels, they're cation channels, that directly couple ligand binding to ion flux. And each insect species has its own specific set of olfactory receptors, or ORs, which can range from just a few receptors, for example in dragonflies, to many hundreds in social species such as ants and bees. And just for some context, we have around 380 olfactory receptors, so the olfactory capabilities of many insects rival our own. What's striking about these ORs is that they are remarkably divergent, both within a species and across species, typically sharing less than 20% amino acid identity. And so here I show a few sequence alignments of ORs from the fruit fly and the Anopheles mosquito, and you can see very few blue amino acids, which would be sites of conservation. And the ORs are so diverse, in fact, that orthologous receptors, those with expected similar odor binding specificities, are rarely apparent between insect orders. So it appears then that each insect has its own unique repertoire of receptors, and this diversity suggests a mechanism that would allow insect species to detect the chemicals specific to their particular ecological niche. Well, one thing that's unique about insects is that this entire complement of ORs interacts with just a single co-receptor called orco. So each olfactory receptor is actually a heteromeric channel where the OR subunit provides odor and specificity to the complex, and the co-receptor orco we think confers structural stability, potentially relaxing evolutionary constraints on the OR and facilitating the evolution of receptors with distinct chemical tuning. So, but on first glance, orco looks just like any other OR, and in fact, the first orco identified was called OR83b, found in Drosophila. Despite looking like any other OR, orco is special because it is the only member of this family that is conserved across species. Many insects have a single readily identifiable orco that's conserved. It's conserved throughout the protein. And in addition to amino acid sequence conservation, orcos actually show functional conservation as well. For example, an OR from one species can seamlessly couple with the orco from another, despite their evolutionary separation. So here I'm showing an in vitro assay where we have co-transfected a mosquito OR with orco, and the response to odor is the same, whether it is paired with its own orco or that from another species, in this case a wasp. Now, orco's ubiquity therefore provides an opportunity to interfere with the entire olfactory system of an insect by just targeting this one receptor. However, orco has been evolutionarily selected not to be activated by odorants, and we have tried, but so far have not been able to find a volatile molecule that activates orco. So one question I'd like to try to address is, why is Orco silent? Given the high degree of sequence diversity among ORs that bind odorants, it's not clear why Orco cannot bind any odorants. Now, although all volatile molecules don't seem to activate Orco, there are a few non-volatile synthetic chemicals that can. Now, we call these types of molecules insect confusants because they don't necessarily repel insects, but rather scramble their olfactory coating. And so in the second part of my talk, I will try to understand how one of these molecules overcomes the evolutionary pressure to unsilence Orco. Now, given the promiscuity of Orco, it is perhaps not surprising that Orco can form a complex with itself, yielding homotetrameric ion channels. These are perfectly functional ligand-gated ion channels, and we view Orco homers as sort of a prototypical olfactory receptor and use as a template for this sort of superfamily of chemoreceptors. Now, I started this project as a postdoc in the Ruta lab at Rockefeller, where I was able to purify Orco in detergent and in complex with monoclonal antibody fragments. And we were able to obtain micrographs of monodispersed particles embedded in ice, as shown on the left. And these are little individual Orco particles where we can determine the structure of Orco by single particle cryo-electron microscopy. And we incorporated monoclonal antibody fragments really to help align our particles. And we could generate nice 2D classes averages that showed clear density for the transmembrane helices and the FABs bound to Orco. And through single particle cryo-EM, we determined the structure of Orco. And as expected by the lack of sequence homology with any other family, Orco's architecture really represents a novel fold. So each Orco subunit has 7 transmembrane spanning helical segments, with S1 through S6 projecting out into the lipid membrane, isolated from other subunits. And there's really only a single helix, this S7 in red, that lines the ion conduction pathway at the center of the channel. But as a consequence of this architecture, there are really actually very few intersubunit interactions within the plane of the membrane. So I've highlighted residues within 5 angstroms of a neighboring subunit, and you can see that the majority of the intersubunit interactions reside in this sort of small cytoplasmic helical bundle, which we have termed the anchor domain. And this is perhaps better appreciated by looking at cross sections to the protein, which I show on the right, where as you can see near the extracellular surface, there's just a few interacting residues in between subunits, in contrast to sort of a dense network in the anchor domain. We think that the tetramer then can be sort of coarsely divided into sort of 2 types of domains. Sort of these 4 loosely attached peripheral transmembrane domains and a central tightly packed anchor domain that moors the 4 other subunits together. Now, one implication of this architecture is that the tightly associated anchor domain occludes the central ion conduction pathway at the intracellular side of the membrane. However, ions do not need to traverse the anchor domain, as 4 equivalent lateral conduits are formed at the interfaces between subunits, and these provide a continuous passageway for ions between the central vestibule and the cytosol. And these conduits are large enough to allow hydrated ions to circumnavigate this tightly packed anchor domain. So it appears then that Orco uses a distinctive quadrivial pore with a single extracellular entryway connected to 4 equivalent cytoplasmic outlets. Now, the narrowest portion of the pore is near the extracellular side, where it is tapered to around 2 angstroms in diameter by a pair of conserved hydrophobic residues. Now, as this narrowing is too small to allow hydrated ions to pass, the structure of Orco appears to represent a closed state. And this is somewhat expected because we didn't add anything that might activate Orco. But one thing that we learned when we were studying Orco is that the lipid membrane seems to play an important role in stabilizing tetramer. So when we extracted the Orco proteins from the membrane using just detergent, we found that the tetrameric architecture would quickly fall apart. And as you can see on this native gel on the leftmost series of lanes, by the addition of just the detergent LMNG, we see the tetrameric species represented by the top band quickly falls apart. But when we add membrane components, either a cholesterol derivative, the CHS, or a mixture of phospholipids, this BPL, we can see that this lipid, this tetrameric arrangement is maintained. Now, given that the potential importance of lipids to the structure and function of olfactory receptors, we established conditions to incorporate orco into nanodiscs. And so nanodiscs basically have a membrane scaffold protein that surrounds orco plus lipids. And what it does is it allows membrane proteins to be reconstituted in a context that mimics a membrane bilayer. And so we were able to develop a preparation that yielded similarly pure and stable protein, but in this case, the detergent molecules surrounding orco have been replaced by phospholipids. And so we were able to determine the structure of orco to a slightly higher nominal resolution of around 2.8 angstroms. And the structure in orco and detergent or nanodiscs is almost identical. But now we observe layers of well-resolved sort of columnar density surrounding the channel in both leaflets. And these density segments really mark the boundaries of the hydrophobic portions of the bilayer. And the membrane layer can be appreciated if we look at it sort of a slice of density through one of the leaflets, where you can see a series of these little gray balls, each one marking sort of a position of one of the lipids. Now, in particular, lipid tails really fill this crevice in between orco subunits and contact the pore. And this packing of lipids between subunits is reminiscent of the voltage-gated potassium channels and trip channels. And we think lipids in this region might help stabilize the quaternary structure of orco, providing a possible mechanism for the requirement of membrane components to maintain the tetrameric assembly. And I'd just like to point out that one of the motifs that really describes the chemosensory families is this TY series of hydrophobic residues QF motif. And this, one of the lipids actually interacts with both the glutamine and the tyrosine of this segment. So these lipids might have some functional roles as well, which we're currently investigating. Now, we did expect to observe density for lipids surrounding the channel, but one surprising observation was density for a lipid molecule inside of the extracellular ligand-binding pockets of each orco subunits. And if we compare our structure of this lipid-bound orco to an odorant-bound OR, we see that the lipids and odors occupy very similar locations. And so density for this lipid is really as strong as the surrounding protein. And the glycerol backbone and head group are really clearly defined and really sit in this extracellular pocket. And we don't know the precise identity of this phospholipid, but the density is consistent with an ethanolamine head group, which is the major lipid type we use to form the nanodisc. And we can see that these lipid tails actually extend out from the binding pocket between the helices S3 and S6. And there's additional space for the lipids created by the presence of smaller amino acids, the serines, glycines, and alanines, creating sort of a space between S3 and S6. In contrast, there's really no passage to the membrane that's apparent from the odorant binding pocket in OR5. There's also large hydrophobic side chains, methionines and leucines, that seem to contribute to a tight interaction between S3 and S6. And similar larger amino acids are more prevalent among orcos, more prevalent among ORs than orcos. And so we think that this tighter interaction causes the S3 and S6 helices to be closer in ORs. So it was really this presence of this extracellular pocket that really got us wondering why orco was silent, since it appeared structurally at least to be capable of binding small molecules. And so we think that the membrane components actively avert spurious orco activity by occupying the ligand binding site and preventing orco from actually interacting with any of the odorants. So now that we have some idea of the challenges to activating orco, we are now in a position to investigate the molecular mechanisms by which confusants can overcome these. And there's essentially sort of two possible mechanisms. Either these synthetic agonists are able to displace the lipid and activate orco, or perhaps they bind elsewhere in the protein. So several synthetic agonists have been identified. The first one is VUA1, which was identified at Vanderbilt University. Greg Pask was actually one of the principal people involved in its identification. And we had investigated several of these agonists, which I should point out activate orco both as a homotetramer and in the context of an OR. We investigated several of these, and we actually settled on studying a related compound called VUA4, which is just a higher, more potent version. And we were able to determine the structure of orco bound to the agonist VUA4 and obtained a similar quality structure of the complex. And we could readily identify density for VUA4 bound to orco in the middle of the membrane. And if you look down at the channel from the extracellular side, you can appreciate how VUA4 wedges itself between helices that line the central pore. Now, importantly, the phospholipid is still present in the extracellular pocket of orco. And in fact, density for almost all of the phospholipids seem unperturbed by the addition of VUA4. But the VUA4 binding this location provides an explanation as to why these molecules can activate orco. They do not displace the phospholipid, but instead they're poised to directly contact the pore and override any ligand inhibition that might occur elsewhere in the protein. So now, while VUA4 appears to be in a perfect location to alter the position of the JAML gate, we actually find that the structure of the ion conduction pathway is still closed. The hydrophobic gate at the extracellular surface remains shut, and in fact, the entire pore is almost identical to the orco structure in absence of ligand. So one reason the pore is closed could be due to the low probability that orco opens even when bound to an activating ligand. Alternatively, our nanase preparation might constrain the ion channel and prevent pore opening. And these are some of the ideas we're currently investigating. But our single particle cryo-EM structural studies of orco, we think, have really provided us with insight into why orco remains inert, even in the presence of odorants, thus ensuring the fidelity of olfactory coding through the family of ORs. We see that the extracellular pocket in orco is actually filled by phospholipid, which suggests that the membrane is playing an active role in keeping orco silent. So we don't know whether other mechanisms also hinder orco, but there are countless molecules in the chemical landscape, and being actively inhibited by the cell membrane seems to be a very robust method to reduce spurious activation. In our structure, the VUA4 bound orco, we think, is really a critical precedent to understand how synthetic confusants can overcome evolution and bypass the canonical activation of ORs or by the extracellular pocket to activate orco by directly interacting with the channel pore. And so we're now in a position to leverage this information to try to design more effective molecules for preventing the spread of deadly insect-borne diseases. Now, I started this project as a postdoc in Vanessa Ruta's lab at Rockefeller University. I'd really like to acknowledge several of the people who have been helpful. Notably, Josefina did a lot of electrophysiology on orco. Martha, Jackson, and Kenzie all performed various activity assays and supported this project in many other ways. I'd also like to thank Tom Waltz and his then postdoc Kelly Kim for incredible help with learning cryo-electron microscopy. And together, Vanessa's lab and I are continuing to work on orco with the Tri-Eye Therapeutics Discovery Institute in New York to derive new molecules that to modulate orco activity with the hope to deter mosquito host-seeking behavior. And I would also like to thank members of my lab who are working on a broad array of projects aiming to understand the basic principles of chemoreception. And so we're also supported by funds from the Gates Foundation. And I'd like to thank you all for your attention, and I would be happy to answer any questions.
Thank you, Joel. Thank you very much. Fascinating presentation. Are there any questions from the floor?
Not yet.
Not yet. Okay. So as you prepare that, I might ask one question here. Then we observe all the time that when we express the receptor together with orco, the response to the VUAI 1, 2, 3, or 4 it's always stronger than when the orco is alone. it's not a completely tight process, but it's a rule of thumb for give us some guidance where sometimes we have some receptors that don't respond to anything, and then we test it to see if that receptor was expressed. It's somewhat related to the question that Marcel asked me during my presentation before, and I don't have a good answer for that anyway. So do you have an explanation why when they are combined, I mean the heteromeric, that the response to VA1 is stronger?
Actually, so we see a little bit the reverse. We generally see,
right?
But that's in a heteromeric system. I don't know what would cause that. It certainly could be that the actual ion channel conductances and things like this are different. We think that because the pores formed by the OR and ORCO, that means the ion conduction properties are going to be probably unique to each channel. And I'll also say that the expression levels, at least in the heteromerics, the heterologous systems that we use, they're very different when you have ORCO by itself versus ORCO plus ORs. And so it might just be related to the stability of the ORCO homomer versus a heteromeric species in oocytes, so that there might be more.
I see that you already answered a question privately to Greg Pask, but it may be for the benefit of everyone. Would you expect a heteromeric OR complex to have a similar wide vial pore or two intracellular exits?
Yeah, this is actually a really good question, because we have this model for the tetramer, and how I like to view it is one or more of the ORCO subunits would be replaced by an OR in a natural olfactory receptor. My favorite model is probably a 2+2, just because I think nature likes symmetry. But what this would mean is that the 4 exits actually aren't the same, because they become from half of it would be the OR side, half of it would be the ORCO side. And so we don't really know what that means for natural receptors. It would mean that they have 2 types of intracellular exits. And so it might not be that they actually have 4 functioning exits. It could just be that 2 of them are. But we expect at least 2 of them would be functional. And from some preliminary mutagenesis, it seems like if we try to mutate in this region, we make very unstable channels a lot of the time. So I wouldn't be surprised if all 4 exits are used, but I think we'll just need to see structures of the heteromeric species to really be sure.
Zain wrote here on the chat, if there is no question from the floor there in Kuala Lumpur, science can be so built. Yes, there's a question there.
Yes, this one, Marcelo.
Yeah, let's hold this one for later then.
Yeah.
Let's give the floor there.
For that talk, I was curious about the lipids that seem to have a very fundamental. Do you have any? They are incorporated to the protein already when it is used in the cytoplasm, or whether they are just incorporated when reaching the membrane?
I think would benefit from a repeat of this question, right, Joel?
Yeah, if I heard you correctly, you're asking if we knew the lipid comes from the cytoplasmic membrane, or if it comes from the lipids that we added during the preparation. Is that correct?
Well, actually, in the real cell, if the lipid are already incorporated in protein when it is used in the cytoplasm, or if it gets the lipids when it reaches the cell membrane.
My bet would be that as soon as it's translated and folded in the membrane, it probably picks up a lipid. So this is a little bit of an artificial system right now. We've reconstituted it. So we've developed some ways to try to pull ORCO out of fruit flies that hopefully we can then see if lipids are there naturally, what the identity of the lipids might be. And maybe address some of these questions about where the lipid comes from, basically.
Would you say that there are 2 different bind pockets, one for the VOL3 and the 4 and the lipids, right?
Yes, yeah. So the lipid sits in what I'll call a canonical odor binding site, and VUA4 is sort of in a new position.
So it goes back to that issue that there are multiple binding pockets predicted, right? Depending on the ligand. Would you agree with that?
Yeah, certainly. So the VOA pocket could be used by an odorant. Certainly it's sort of a very greasy kind of large pocket. But there are lots of, kind of large greasy molecules, especially pheromones and things. So we don't really know if they would use something more closer to the pore, or if they use sort of the canonical odor binding sort of region. We'll have to just see. Certainly this suggests that at least there are multiple ways to open or to activate the channel. And I wouldn't be surprised if nature somehow took advantage of that.
We had similar problem with pheromone binding proteins 15 years ago, but those are detecting lipid-like compounds, right? Pheromones. So that makes more sense. Anyway, reading the question from Zain here, science can be so beautifully explained and illustrated. Thank you so much. In case you have not wondered, is DIT doing something similar to VA4? I do realize that there is no structural similarity. However, from various aspects, DIT appears to mess up Orco. What's your comment on that?
My comment on that is, I don't know how DIT works. It definitely could do this. I find it a very hard molecule to think about, and it's something I would just say I don't know, but I would really like to find out. I think DIT might be doing a lot of weird things, and we're only beginning to understand how it works.
Well, if you would stay, you might be able to take some more questions, but I think in the interest of time, we have to move on and move to the next presentation. That's the closing remarks by May Berenbaum. Let me move the slides here and start sharing the slides. Closing remarks by May Berenbaum. If someone does not need an introduction, it's May Berenbaum, but in any case, I'll let the White House do that.
May Berenbaum. National Medal of Science 2, May Berenbaum, University of Illinois at Urbana-Champaign, for pioneering studies on chemical coevolution and the genetic basis of insect-plant interactions, and for enthusiastic commitment to public engagement that inspires others about the wonders of science.
In case you don't know that person, May Berenbaum is Barack Obama. May, the floor is yours.
Okay, I'm unmuting and starting video and asking you if I still have 10 minutes because we're running.
Yes, no, go ahead.
Okay, good. All right. Well, this is the end here. We all owe an enormous thank you to the indomitable Walter Leal, who I think, given the opportunity, could bring people together and bring about world peace. This accomplishment, this symposium spanning 48 countries is truly remarkable, and an example too of how science communication has changed in so many fundamental ways. So for some of the speakers in today's Frontiers of Chemical Ecology, and shout out to BedoukianBio for supporting this wonderful effort. Some of the speakers today, the Rising Stars Symposium, this might have been their first scientific meeting. If you could, next slide. Okay, so I am the opposite of an early career investigator, so I thought it might be interesting or informative to describe how one progresses from early career to don't even ask me how long I've been doing this. I attended my first scientific meeting in February 1980. It was the first Gordon Conference on plant-herbivore interactions. As I attended as a graduate student 6 months before defending my thesis, I was invited to serve as a panel discussant, not because I was a rising star, but because Paul Feeney, my advisor, was co-organizer of the meeting, and he brought along everybody in the lab. This is us, I think, en route from LAX to Santa Barbara. In fact, the meeting was a week before my job interview at University of Illinois, where in August I actually began, and I've been there ever since. Yes, 42 years. In a way, this Gordon Conference was a prequel to my first International Society of Chemical Ecology meeting in 1984. Next slide. This is the Gordon Conference. It was mind-boggling. It was not like other conferences, most other conferences, which are hundreds or thousands of people and concurrent sessions. The structure of a Gordon Conference is basically scientific talks, a break in the afternoon for interaction and conversation, and then eventually posters in the evening. I was totally blown away by this. You can see me, the red circle there. Not only am I standing next to another early career investigator, one of Paul Feeney's other students, Mark Rauscher, who's now the John Carlisle Kilgo Distinguished Professor of Biology at Duke University, but look who I'm behind. I'm standing behind Dan Jansen, who at the time was a legend. Every legend you can think of in chemical ecology was at this meeting. Tom Eisner is there. Gottfried Frankel is in the picture. Vince Tatier in the front row. It was just an incredible experience and an unforgettable one. Next slide. Okay, but more appropriate to ISCE was the second Gordon Conference. That was in 1983, and I attended that one as well. And that was held in the Holiday Inn in Ventura, California. I did actually, I was invited. By this point, I was a faculty member. I got invited to give a talk. But probably the most interesting aspect personally to me was the fact that the venue, right near the beach, was only 3 miles away from an overwintering site for monarch butterflies who undergo this heroic migration. There across the beach were hundreds, if not thousands, of monarch butterflies being eaten by the feral cat population at the time. And poor Lincoln Brower, who demonstrated why they were unpalatable and indigestible, had to explain to every member of the Gordon Conference attending the Gordon Conference how cats could get away with eating these things. More to the point and more important, next slide, for the history of chemical ecology, it was at the Ventura Gordon Research Conference that the decision was made to found the International Society of Chemical Ecology and adopt the Journal of Chemical Ecology as its official scientific society publication. You can see the history. It's posted on the website of the ISCE. This is really a foundational meeting. Next slide. This is where all the concepts and principles and goals and missions were laid out. The first ISCE meeting I attended was its second meeting. I didn't go in 1984 to Berkeley, but in 1985 I went to Madison, Wisconsin. That's the program from that meeting. I didn't give a talk, but by 1986, just to show how rapidly things can change, I was offered the opportunity to edit an entire special issue in Journal of Chemical Ecology. That meant a lot to me. To have this opportunity, basically at the time my main interest was in photobiology and photochemistry and their role in plant herbivore interactions. So this, I still have in my office, this special issue, yellowing, decrepit original issue is still in my bookshelf because that's how much it meant to me to have this opportunity and become more of a recognized contributor to the discipline. Next slide. So almost 30 years after that is a way of expressing my gratitude to International Society for Chemical Ecology. By that point, I was a department head and I had offered to help celebrate the 30 years of ISCE existence by hosting the annual meeting at the University of Illinois. So why go to a meeting? It's an opportunity to see old friends, meet new friends. There's my advisor on campus with me and the revered Jim Tumlinson, whom we lost just recently and who was called out during Erika's talk during this session. Next slide. And here I am now at the end of the program, offering, I hope, some useful information and maybe some inspiring thoughts as requested by Walter. Next slide. Next slide. Okay, so what can you expect from participating in a scientific conference at any age or any level of experience? Meeting formats change. The Gordon Research Conference itself was invented in or founded in 1930 and was a great departure of from meetings at the time, which were large, relatively anonymous gatherings with relatively few social opportunities for social interaction during the meeting itself, whether there were pre or post social activities. Another thing is that methods evolve. The methods that are so important now to early career researchers will soon be replaced by better methods, which will enable you to answer hitherto intractable scientific questions. Scientific questions will come and go, and your scientific identity will likely change with them. And beyond the scientific content, there will be conversations, camaraderie, collaborations, and enduring friendships. Next slide. And so I think this unique session provided a great illustration of what early career researchers mean to a discipline and what it means to be at the frontier. Probably the most impactful message is that there are all kinds of frontiers. There are methodological frontiers, conceptual frontiers, and even motivational frontiers, why people are undertaking certain research projects. And it seems to be the case that frontiers attract early career researchers. And that's actually a very good thing because they are free from many biases. They are not baked into or indelibly linked to what they've learned before, and they make change possible. And in fact, I could not have asked anyone to start off this symposium better than Greg did when he mentioned the story of the lesson of which was, don't always listen to your principal investigator, because having a student who didn't exactly pay close attention, but instead made a suggestion, basically changed the focus of the work, at least the organism involved in his project on ant chemosensory sensilla microstructures. I also really appreciated hearing about, after 2.5 years of pandemic and no contact deliveries, hearing about touching and contact deliveries for chemical signals among insects. So Nicoletta, as Walter called out, was demonstrating a venerable principle in chemical ecology, that's chemical prospecting. That goes back to the founding in 1983, the founding principles, where research areas were spelled out as including the chemistry, biochemistry, and function of natural products, their importance at all levels of ecological organization, their evolutionary origin, and their practical application. But importantly, a new frontier illustrated by Nicoletta and several other speakers is that the practical application can be mechanism guided, knowledge guided, rather than guided by hope, optimism, and a good deal of luck. Often trying to identify useful natural products simply involve making collections in tropical forests, bringing back extracts, and then just hoping for the best, or pulling synthetic chemicals off the shelf and just testing them in a random fashion, which was actually a path to very low success rate, enormous expenditures of time, energy, and resources with very little notable but few successes. And mechanism-guided searches for practical applications are the promise for the future. Erika, on her part, looking at the curious response of the black-legged tick, similarly used a different approach rather than random trials of chemicals that were either already available or extracted, embedded her study in the community rather than doing lab-based work without the complexities introduced by biodiversity and a community context. It's really impossible to get to the mechanisms. Trevor, working on a persistent behavioral state in mosquitoes for pursuing humans, took advantage of a methodological significant advance using optogenetics, activating neurons but with light. This method is relatively new. In 2010, it was called Method of the Year by Nature Methods Journal and the Breakthrough of the Year by this journal Science. Optogenetics, classic new technological advance, allowed people to develop approaches to find answers to hitherto unanswerable questions. Chloe, on personal care products, altering mosquito host interactions, illustrates the frontier of anthropogenic chemical ecology. So this is recognition of the fact that we humans use natural products, and by our use of natural products, we affect species interactions. This is a factor influencing certainly the ecology and eventually the evolution of chemical signaling, and also promises to have big payoffs in the future. Ali, looking at species-specific differences in mosquito olfactory responses and repellent, again, like Nicoletta and also Erika, using a mechanism-based search for practical, useful materials. Just want to make the point that Joel briefly addressed. DEET is still the standard repellent. DEET was discovered in 1946. Its properties were discovered in 1946 in one of these random searches, and it's been registered for public use in 1957. We don't have many alternatives to DEET that are as effective, and we still don't exactly understand how DEET works. So this is a frontier. And finally, Joel was at sort of a conceptual frontier and also a methodological one, using, as he did very creatively, the cryo-EM, which was in 2020 declared by the journal Nature revolutionary for 3D structural elucidation. As a conceptual frontier, the idea that insects can have this fundamentally different mechanism for olfaction from any other organism was very slow in coming. And I know this to be a fact because I taught chemical ecology for many years at University of Illinois. And that's the one lecture that year by year by year, I had to keep changing because it took a long time for this realization to be embraced and then to restructure how olfaction in insects mechanistically works. That's really, I don't know if I went over time. If I did, forgive me. But just let me conclude by saying thank you, early career investigators. I have to say I have a different perspective listening to this meeting as I did for my very first International Society of Chemical Ecology meeting. Now I'm a department head and I keep listening to these talks thinking, how can we steal these people away from their institutions so that we can hire them and have them in our department? So wishing you all what seem to be very likely productive, fruitful, and lengthy careers that will inspire us all. So thank you for listening. I don't know that you need any questions. It's pretty late where some people are, it's 11:43 where I am. So, and I'm clearly at home. And I don't know, Walter, what's next?
Thank you very much, May. May, if I could ask you one question, can you identify the gentleman, please, in this video?
Oh, I did not know there would be a test after this. Paul, that's Paul.
It's Paul Finney.
Yeah, what?
And let's see what Paul Finney has to say about you.
Oh, you did.
Absolute, one of the most well-known students of mine is May Berenbaum, who's everywhere all the time. She's a very distinguished scientist, very tremendous intuition, an amazing memory, not just for science, but for all kinds of miscellaneous stuff, including the best desserts to get out of food machines. She was here, all kinds of trivia. I can remember in lab sessions, in the lab, people would be working away at their projects, the radio would be on in the background, and there'd be a quiz show, and the prize would be, 2 tickets to a movie at the local cinema. And May was always the first to the phone, instantly, when asked what actress played what role in what movie in 1938. She'd be there. And she was foiled time and time again by the slow communication between the Cornell telephone system and the real outside world, which meant she was very rarely successful in actually getting the movie tickets. But May went on to great things. She was elected, one of the youngest women to be elected to the National Academy of Sciences pretty early on in her career. Her research was incredibly rigorous look at coevolution going on between umbelliferous plants and insects, various kinds of swallowtails and various other species, both here and later in Illinois. But she also contributed ideas in a whole range of different things I do not mention now. And she was an amazing stimulator of everyone else around her. So in that sense, she also had an outsized contribution to my lab.
He's one of my heroes. Oh, I don't know how you did that, Walter, but
no one knows that I have this video about people, okay? I want to surprise you. That was a very nice
very
segment.
nice.
There.
I was not expecting that at all.
Everyone in the panel, feel free to come over with your camera and the video. I would like to thank everyone for attending. I would like to thank Bedoukian for sponsoring. Also all the speakers for the participation. Also the technical people from Pacific Green Concepts that work together to team up here. Leanne, Javis, and Mona. It was a pleasure to work with you. We will be back in 2 days with the second part of the Frontiers in Chemical Ecology with 80 speakers. ISCE-APACE will continue after lunch break. Those attend the ISCE-APACE meeting remotely, please make sure to use the link provided via email by Pacific Green Concept for the rest of the day. Okay, so for everyone else, good night, good morning, or good afternoon, depending on your time zone. Thank you very much. Thank you very much all that make this wonderful symposium, all the speakers here. Again, feel free to, if you want to say something here before we end. Anyone else? That was an amazing closing remarks by Berenbaum, talk specifically about each particular speaker and give a nice history of chemical ecology at the closing. Thank you. Thank you all.
Thank you.
Thank you. Thank you.
Thank you.
Thank you.
Thank you, and thank you, Walter.
I made a tweet. I made a Twitter tweet, so you can follow the thread. So everything's there. All the pictures are there, Walter. That'll make everyone happy.
That's